seems to work this time.
Hello, Professor Sil. Did you join us already?
Yes, I think it works now.
Okay, thank you.
Oh, I saw my video doing funny things. Yeah. But when I share my screen, let's see if that works. Is that okay if I try?
Yes, but please consider that we already accept some participants, so
they can see it.
Yes, they can see it. If you want to present some surprise, then it won't be it won't be a surprise anymore because some participants already attending here. And probably the video is again.
Yeah, I see it. It's not good. I don't know why that's happening.
So did you need us to share the screen for you?
Wait, let me, because I think when I share the screen, you should be able to see it. Let me just try.
Okay. Mas, bisa dibantu nggak?
I cannot share at the moment, I think. Because another participant is sharing. Okay. No, yeah.
Yes, just wait a minute. Please be patient. Wait a minute.
No worry.
Jangan di-mute kan? Hello, Professor Sil. Can you try now to share your screen?
Okay. Can you see that?
Yes, we can see that. Biodiversity conservation in Southeast Asia, right?
Yep. So it's just my video doing funny, right?
Yes. Yes.
Okay, yeah. I think something with the driver of the camera, because this has happened before.
Oh.
Let's see. Oh, can I try? I don't know how this works when I do full screen.
Yes, yes, you can. Of course, you can try it.
Oh, how does that work? Oh, yeah. Okay. Fine.
Yes.
That looks nice. Okay, so everything works except my video.
Unfortunately.
But I can try to switch it on every now and then.
Yes.
How do I do that?
Just press the pitch down button also can work.
Okay. Right. Okay, so I stop sharing the screen, right?
Okay, yes. Thank you very much. So we still have some time before your session.
Yep.
Yes.
I'll just wait.
Oh, thank you very much.
Oh, so Indonesia still has the virus?
Of course. How about in Brunei?
Yeah, we got rid of it in April, so it's completely free here.
Really?
Yeah.
Wow.
We're lucky.
Because yeah, you are lucky living in Brunei. But in Indonesia, the case is still increasing because we have some holiday, and I guess people is already had stress, so they want to take a holiday. They didn't. They insist to meet their family and something like that, and then there's new cluster with the family, with meetings also, and in the
same things happening in Europe.
Tourist area. Yeah. Yes. So for a whole year, you stay in Brunei, didn't go anywhere.
Oh, yeah, that's the problem. We can't leave the country. Well, we can, but when we come back, we have to go into quarantine for 2 weeks. Yeah. But Brunei is a small country, so it's more easy to control who goes in and out than a country like Indonesia.
Yeah. How about the vaccine development in Brunei? They're not doing.
Not yet.
Not yet.
I haven't heard about that yet.
Oh, yeah, because it's already clean in Brunei, right? So is it possible for the foreigner to come to Brunei?
Yeah, I can still come, but they will put you in a hotel room for 2 weeks, and you cannot leave the room.
Oh.
Before they let you out.
Okay.
So almost no one is traveling anymore. Everybody stays in the country.
Yeah, that's more safe for now. Also, welcome to all the participants. Professor Slick, I would like to introduce you. We have the head of Research Center for Conservation, or previously the head of Bogor Botanical Garden. There is a Professor Dr. Hendrian. Hendrian, would you like to say something?
I know
yeah,
Hendrian.
of course. Pak Hendrian, would you like to? Center for Conservation.
Hello.
Or previously the head of Bogor Botanical Garden.
Long time no see.
Dr. Hendrian.
I heard that you're in Brunei now.
Would you like
I think
to
I know.
say something?
I know.
Yeah, of course.
Yeah, I want to visit Indonesia, but it's not so easy to leave
would you like
Brunei
to?
at the moment because of the quarantine.
You're very welcome.
Center for
A little
Conservation.
reunion here.
Or
Hello,
previously
Barry.
the head of
Would like to come.
Botanical
Long
Garden.
time no see.
Dr. Hendrian.
I heard that you're in Brunei now.
Would you like
I think
to
I know.
say
I know.
something? Yeah, of course.
Yeah, I want to
Yes.
visit Indonesia, but it's not so easy
Pak
to
Hendrian,
leave
would you like
Brunei at the
to
moment
be a good
because
chance
of the
also
quarantine.
to gather
You're very welcome.
old friends?
A little
Conservation.
reunion here.
Or
Hello,
previously
Barry.
the head
Very
of
nice nice place.
I'd like to come.
Long time no
We
see.
have also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you
I
like
think
to
I know
visit there?
the flexibility.
Oh, of course. Did you know her?
Yeah, I want to
Yes.
visit Indonesia, but it's not so easy to
Perhatian
leave
for
Sweden
maybe a good
because
chance
of the
also
quarantine.
to gather
You're very welcome.
old friends.
A little reunion.
Hello, Mary.
Very nice place.
I'd like to come.
Long time no
We have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you
I
like to
think I know
visit there?
the flexibility.
Oh, of course. Did you know her?
Yeah, I want to visit Indonesia, but it's not so easy to
Perhatian
leave
for
Sweden
maybe a good chance
because of
also
the
to also
quarantine.
gather
You're very welcome.
old friends.
A little reunion.
Hello, Mary.
Very nice place.
I'd like to come.
Long time
We
no
have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you
I
like to
think I
visit
know
there?
the flexibility.
Oh, of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's
we're
not so easy
we're
to
happy
leave
to
Sweden.
visit you early.
I want to
We
make
have also
sure.
gather
You're welcome.
old friends.
A little
So maybe
reunion.
now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
I don't
Long
know.
time no
We have
see.
also director
Your
of the
place,
Center
right?
for Biology.
I heard that you're in Brunei, no?
Would you
I
like to
think I know
visit there?
the flexibility.
Oh, of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's not
we're
so easy
we're
to
happy
leave
to
Sweden.
visit you early.
I want to
We
make
have also
sure.
gather
You're welcome.
old friends.
A little
So maybe
reunion.
now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
I don't
Long
know.
time no
We have
see.
also director
Your
of the
place,
Center
right?
for Biology.
I heard that you're in Brunei, no?
Would you
I
like to
think I know
visit there?
the flexibility.
Oh, of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's not
we're
so easy
we're
to
happy
leave
to
Sweden.
visit you early.
I want to
We
make
have also
sure.
gather
You're welcome.
old friends.
A little
So maybe
reunion.
now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time no
We have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to
I think I know
visit there?
the flexibility.
Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but
we're
it's not so
we're
easy
happy
to
to
leave
visit
Sweden.
you early.
I want
We
to
have
make
also
sure.
gather old friends.
A little
So maybe
reunion.
now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time
We
no
have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but
we're
it's not so easy
we're happy to visit you early.
I want
We
to
have
make
also
sure.
gather old friends.
A little
So maybe now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time
We
no
have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's
we're
not so easy
we're happy to visit you early.
I want to
We
make
have
sure.
also gather old friends.
A little
So maybe now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time no
We have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's
we're
not so easy
we're happy to visit you early.
I want
We
to
have
make sure.
also gather old friends.
A little
So maybe now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time no
We have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's
we're
not so easy
we're happy to visit you early.
I want to
We
make
have
sure.
also gather old friends.
A little
So maybe now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time
We
no
have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's
we're
not so easy
we're happy to visit you early.
I want
We
to
have
make sure.
also gather old friends.
A little
So maybe now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time
We
no
have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's
we're
not so easy
we're happy to visit you early.
I want to
We
make
have
sure.
also gather old friends.
A little
So maybe now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time no
We have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No,
but it's
we're
not so easy
we're happy to visit you early.
I want to
We
make
have
sure.
also gather old friends.
A little
So maybe now I just
Hello,
wait.
Mary.
Very nice place.
I'd like to come.
Long time no
We have
see.
also director of the Center for Biology.
I heard that you're in Brunei, no?
Would you like to visit there? Of course.
Yeah,
Did you know her?
yeah, I want to visit Indonesia,
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, just our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, our friends. Also, director of violence.
Another few in Brunei.
I want to visit.
No, we're happy to really. But also, our friends. No, we're happy to be. But also, also director of No, we're happy to be. But also,
rindu peternak
also director No, we're happy to be. But also,
rindu peternak
also director No, we're happy to be. But also, also director No, we're happy to be. But also, also 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 No, we're happy to be. But also, also
keanekaragaman
No, we're happy to be. But also, also No, we're happy to be. But also, No, we're happy to be. But also, No, we're happy to be. But also,
Meti.
some quite alarming titles like Southeast Asia Biodiversity and Impending Disaster, Tropical Turmoil, Biodiversity Tragedy in Progress, and Momentum Drives the Crash, Mass Extinction in the Tropics. You can actually also see that in these kind of maps. I think many of you may have seen these maps. I still like to use them, although they are a little bit outdated. But they show very clearly what kind of problem we are facing. So, for example, if we look at Southeast Asia, mainland area, for example, in 1970, and I always like to give the example of, for example, an elephant or a tiger. If you were an elephant or a tiger and you were living here, so almost in China, you could still walk through the forest all the way to Malaysia. Now, I don't say that that happened, but it was possible because most of the forest was still connected. So it also meant that the genetic exchange was still more or less continuous. Even though the animals may not have moved all the way from the north to the south, their genes might still have moved freely through the whole region. So you can see by 1990 that is as good as impossible. All the forests has been fragmented and all the forests are like little isolated patches. So they have become independent of each other. And the same thing is happening here in Sumatra and also in Borneo, where most of the lowland forest has disappeared over time. And at the moment, New Guinea is also a hotspot of change. Although this 2020 map was a projection, and I think actually currently it's not as bad as shown in this picture. Anyway, but this picture clearly shows what the problem is that we are facing. Now sorry for my video, which is doing a bit strange. The main driver of this crisis is just simply human population density and growth. So people are everywhere at the moment. You can look at this graph here at the bottom. You can see human population densities were quite stable until more or less the introduction of antibiotics and modern medicine, and then human population started exploding. And you can see the main driver of most crisis of the population increase. Human population has happened in Asia, and people are everywhere at the moment. You can look at this, which actually means that the tropics in Asia are also were most quite stable in the world until more or less the introduction of antibiotics and all these people they need a good living, human population started exploding, and you can see the main driver of most crises of the population increase, more problems for the population has happened in Asia. Now we have many everywhere at the moment. Things that which actually means that the results from the high human population density is stable. This is, for example, habitat loss, fragmentation, and degradation. I have an example here in a district in China where I used to work. You can see that in 1988, most of the area was still green, and green means forest. And then we have these patches here of pink, which are rubber plantations at that time. This is, for example, you can see by 2003 and 2006, the rubber plantations have exploded and covered almost the whole. You can see that in 1988, and all the forest that was originally there is now fragmented. And actually, and then we have these patches of green that are still there, are only there because they were nature reserves. I'm quite sure that if you can see by 2003, nature reserves, they were converted to the rubber plantations have exploded, rubber plantations as well. The whole area. So when you drive on the road now, most area is now fragmented. If you drive actually, you still see forest, but it's only one species of green that are still there. Are only there because they were nature. That makes it quite a big impact. You can see by 2003, nature high that they were converted to forest with one species plantations have exploded, rubber plantations and the whole exotic. So when you drive on the road now, so this is is a big problem that is happening all over the region. Actually, then we have this problem with fires, which are now almost happening at the frequency of only there a yearly frequency. That makes it as this is an example of East Kalimantan in Indonesia, where we had these big fires in 1983 and again in 1987 so 1988. So this is a big problem. This area indicated in yellow, orange, and red problem with fires, with burned forests or burned areas, which was more than a year and a half million hectares was burned. But the interesting thing is when we look at all these fires, where did these fires happen? So you can see that a lot of fires actually happened in areas where the forest was already gone. So these were this area indicated in yellow, orange, and other fires happened in log forests. So these were forested areas disturbed by people, and then very few for fires. The interesting thing happens when we look in the undisturbed forest. Where did these fires happen? So this already shows what we knew, that actually if you have a closed primary forest, the chance of a fire is actually very large. But as soon as you start other fires, there can be a log forest, so these were forested areas, the forest becomes much more susceptible, and then very few for fires. The interesting thing happens when we look in the fires are not only a result of this, but already shows what we knew, that actually if you have a closed primary forest, the chance of a fire is actually very large. Then on top of that, you start over exploitation of resources, forest, wildlife, and plants, and this is also related to the fragmentation of the forest, because the more the forest is fragmented, the easier it is to actually enter these forests and take animals and plants out of these forests. And Southeast Asia is actually one of the main illegal wildlife areas of the world. So trading areas in the world. This is also something really has to be done here, because the volumes that are traded are really reduced and not actually enter these forests. Another problem that we now have is invasive species. Invasive species in Southeast Asia areas that have already been disturbed. Illegal wildlife here. This is example areas of the world. So Java, trading where this is also something really has to be done. American volumes that are secondary forestry was introduced into the Bogor Botanical Gardens. Probably, we're not completely sure how it spread, but that's a likely source. And you can also see it spread from Bogor all the way almost already to Jakarta. So this whole area, you can now find this exotic tree. Now, this Cecropia is actually not such a big problem. It just mixes with the local species. It doesn't really cause any problems. So in this case, we are still lucky. But there are also examples like this, the water lettuce, which is an invasive also from the Americas, that can cover a whole water body. So this area, what I show here, is not a field of plants. This is a lake completely covered by the water lettuce. Now you can imagine what happens if this plant is covering the whole lake. There will be no more light going to the bottom of the lake. So the whole ecosystem of that lake is completely changed because of this plant. And because it grows so fast, it's also very difficult to get rid of it. So invasive plants, also a problem. Then of course we have pollution itself, like pollution coming from mining, here also gold mining, for example, or people just putting their rubbish in the rivers, and not only people, but also industries, or pollution like here, like from infrastructure and heavy industries. Now, another problem is land use planning and law enforcement. It is actually, in principle, very well possible to develop an area for a specific crop like oil palm or rubber plantation, but still leave forest in between the plantations, so that, and especially if you can do it in a network way, and so that biodiversity can be maintained within this productive landscape. But what often happens in the tropics is that the development of these plantations, even though perhaps they may have been planned well, the execution often results in complete change of the landscape, with the whole landscape covered in this plantation area without any forest remaining. Here I have an example from Indonesia. It's Gunung Palung National Park, which is a very national park because it contains a mountain and it runs all the way to the coast. So it actually contains most of the forest types that you can encounter in Borneo in a single national park. You can see what happened in 1988. So this is the park, the yellow line. The park was still more or less intact, although there was already some human activity here on the east side of the park. So these red parts are areas where the forest has been cleared for mostly oil palm, I think. And this is the situation in 2002. So lots of forest has disappeared around the park, but you can see also inside the park. So this is not supposed to happen. So this is a problem with law enforcement. So this is an example from Indonesia, but these things are happening all over Southeast Asia. So this is also a major problem. And then on top of that, of course, we have forests and plantations will change in the future because the temperature will go up and the rainfall patterns may change. We may get on top of that more intense droughts and plantations will change in the future. So that will also already affect the rainfall patterns may change. We may get on top of that more intense droughts and plantations will change in the future. So that will also already affect the rainfall patterns may change. We may get on top of that more intense droughts and plantations will change in the future. So that will also already affect the rain. Okay. So this was the depressing part of the talk. Let's switch to the exciting part. So while the environment is degrading around us, our possibilities to study this and monitor this over time have increased dramatically. So for biologists, this is a very interesting time because we have now so many possibilities to study the environment. So one of the things that have happened over time is, well, of course, the internet. And well, this whole meeting is an example of that, right? We're talking over internet. So more and more species data have become available online. And I can remember when I was a PhD student, which is not so long ago, so that was around the year 2000, I really struggled to find information of the tree species that I had collected in my fieldwork in East Kalimantan. I had to go through books, ask experts. It took a lot of time. But nowadays you can just type in all these species names and you get information almost right away. So I have some examples here. Maybe you know the Global Biodiversity Information Facility, where you can find plant and animal collections around the world. So it's very easy to see where the species is distributed. There are these very large databases developed now, like the TRI, the Plant Trade Database, where you can just download information that you need for your study. There are now websites where you can see pictures and descriptions of plants, etc. So also several examples from Indonesia. So this is already a great help. A lot of this data is now also used for applications like this, automatic identification tools. So where you can use your mobile phone to take a picture of a plant and the plant will be identified for you, or they will find something that looks similar to it. So it may help you identify a plant. Now, many of these applications work quite well in temperate regions where there are not so many species, but there is still a big challenge for biodiverse rich regions like Indonesia, for example. We are trying to make something like this for Brunei, but even in Brunei, we already have 5,000 species of plants. So it's a huge task, but it's really a nice challenge to try because it will involve people more with nature, right? If you can just use your phone to take a picture of a plant or animal and the phone will give you the information about the species and what it does. Okay, this is also really exciting what is happening. This is actually already quite old, DNA barcoding. DNA barcoding is the sequencing of a very small part of the DNA that is very easy to sequence and very cheap because it's so short region. And they have done this for animals first. Plants is slightly more difficult because for plants you need 3 different regions to get a good identification, but for animals you only need 1. And this really is a big help for several reasons. Well, one, for example, I have an example here. This is one of the first studies where they used DNA barcoding. So this is, I think, already more than 20 years ago, was to find out which caterpillars fitted with which butterflies. That's actually a big problem because the only way to find out, especially in the tropics where there are so many species of butterflies, to find out which caterpillar belongs to which butterfly in the past was to breed the caterpillar and wait what kind of butterfly would come out. But now we can just sample the caterpillars and the butterflies and directly link them together. Well, there are many examples like this. One of the most exciting ones, in my opinion, is that using this technology, we can actually also identify microbes that live in the soil. So suddenly we can identify the bacteria and the fungi living in the soil. So this is something that I would like to do here in Brunei. And I think in Indonesia, there are also many possibilities for this. So you can just take little samples of soil and sequence them and find out what species, so how many species of bacteria and fungi there are and also what species there are. And you can compare between different areas. You can actually also study the roots of trees using this method, because normally when you dig in the soil, you can find the roots, but it's almost impossible to find out what species of plant the roots belong to. But using the barcoding, you can easily find out. So really exciting. Even more exciting now is the genomics, so the next gen sequencing, where we can actually just sequence the whole genome. And these technologies have become so good that it's now even possible to collect a sample of water from a river and still find in that sample the DNA, so that's called environmental DNA, of species that live in that river. So you can do an inventory by just collecting some water. So this is also an exciting new development. But if I come back to that soil microbes example, one thing that we can do with these new technologies is not only identify how many species of microbes there are and what species of microbes there are in the soil, but we can also extract direct information on the genes that are active within the soil. So we can get a functional picture of what is happening in the soil. Because with these technologies, we can recognize genes that are present and active in the soil. Okay, so really exciting possibilities here. One of the other nice things that we can do once we have these DNA sequences is e-taxonomy, which makes the work of ecologists and also taxonomists much easier. So in this example, I have a plant specimen from an herbarium, but it could also be a living plant from a plot. Anything is possible that has DNA. So once you have the sample, you extract the DNA, or you have many examples from a plot, for example. You extract the DNA, you sequence it, you can compare how similar they are, for example, using BLAST. Based on the similarity, you can group them into sequence clusters. So these sequence clusters are species that have very similar DNA. So they are most likely very closely related species within the same genus or family. So once you have that, you can take a look at your actual specimens again and see how well the computer has grouped your specimens. So this can be done very quickly, because normally when you are identifying plants, or even worse, if you are identifying insects, for example, it can take weeks, months, sometimes even years to identify all your material. Well, this kind of technology can do it within, well, several hours to a week, perhaps. So it saves a lot of time. Even if you don't know the name of the species, but you just have these sequence groups, you can still do lots of ecological analysis. You can compare the similarity in sequences between different sites, etc., or the diversity of sequences in different sites. And another thing that you can do is, because this is DNA sequences, you can also use that to produce, for example, a phylogeny of the species in your sample, which is also interesting for biodiversity research. So that's this example, for example. So here we have a tree species diversity map of Borneo that I made quite long ago already. So it may be outdated already, but still you can see that this mountainous region in Borneo has quite low diversity of tree species, while the lowland areas, which are actually also the most threatened areas, have quite high tree diversity. So this is the traditional diversity map that we could make in the past. Now, once we have the phylogeny of all these species, well, in this case it was genera in these areas, and we combine that with the plot information, we can actually turn the phylogenetic information into a phylogenetic diversity map. And then we get this map of Borneo, where actually these mountainous regions show up as the areas with the highest phylogenetic diversity, which means that in these areas you find groups of species growing together that are either very old lineages or they are very distantly related. While in the lowland areas, where you find these whiter colors, so lower phylogenetic diversity, you often find species growing together that are closely related to each other. For example, many species of dipterocarps. So this suddenly gives two different views of the diversity of Borneo, which are useful for conservation. Now, remote sensing is another very exciting field that we have nowadays. We can do really nice things now, and this one is one of my favorites, because once we can do this for the tropics, so this is also very nice for Indonesian to try. We can suddenly do tree inventories for whole very large landscapes, or for example, the whole of Indonesia if needed. So this is based on specific frequencies of light that plants reflect. And it turns out that almost every species of plants, so here we see a list of different species of plants, has their own specific reflectance. So you can actually identify specific species of plants just from a picture that you take with the right frequencies, of course, of the forest. So here there is an example where they have identified the trees in the picture using this technology. So this has some very nice applicabilities, because, well, not only can we map species compositions over very large areas and calculate the diversity for every area using this method, we can also pick out specific species, like here. For example, if we have a problem with an invasive species somewhere, the old way was to first you had to find all these invasive species in the area, and then you could eradicate them. But using this technology, just taking one picture, you can immediately identify where all these plants are. So it makes it much more efficient to eradicate that species, if that is the aim. So really exciting things. I hope I can also do this in Brunei. This is already a bit older, but still being more perfected. This is also remote sensing using radar. So with radar, you can actually measure the height of the ground level and the height of the canopy. And the height of the canopy is often directly linked to the amount of biomass in the forest. So by just taking a radar image here for this mountain in Hawaii, you can immediately calculate the biomass pattern for this forest. Have high biomass, and the areas have high biomass, and the blue areas have low biomass. So if you can do this for a whole country, you can actually very exactly calculate the amount of carbon stored in the forests of these countries. And nowadays, these you also have handheld or ground-based radar technology, so you can actually take very nice three-dimensional pictures of trees using this technology. Okay, most of these remote sensing equipment used to be quite large, so could only use it on planes or on satellites, which made it a bit complicated. But nowadays they have become much smaller, and you can actually fit them on drones. So drones, conservation drones in this case, they are also a very important new development, because it makes it possible for simple researchers like us to use this fancy technology to study the forest. This is a very early example from Sumatra, where they were looking for orangutan nests, and they were still using these planes instead of the, how do you call these, quadro helicopters or something that people use nowadays. But well, the nice thing is you can just program the course for this drone, and it will fly this course by itself and then come back with all the camera images that you want to get. Okay, a very important development over the last few decades is also the availability of long-term plot data. So often people make plots in forests, and if these plots are permanent plots and you keep monitoring them over a very long time, suddenly you become able to see how the forest is changing over time. So this is different from establishing plots just once and then never visit them again. This is really monitoring plots over a very long time. So some of these plots have now been monitored for several decades, and we are finding really interesting patterns. So in the past, people often thought that tropical forests were quite stable environments, but it turns out that they are very dynamic. So one of the things that has been happening in the Amazon region, for example, but probably also in Africa and Asia, is that the lianas have become more common, because it's probably related to the climate change, because we can also see that the tropical forests in South America and also in Africa, and slightly less so in Asia, have been increasing in biomass. So the forests have actually been taking up a lot of carbon dioxide from the atmosphere and turned it into wood. So you can see here from the 1980s to early 2000s, the amount of carbon stored in tropical forests just increased and increased and increased. This is probably because the carbon dioxide concentrations in the atmosphere also increased, and carbon dioxide is a very important resource for photosynthesis. So in the past, probably carbon dioxide had been limiting for tree growth, and because we are adding carbon dioxide, it's actually good for tree growth. But you can also see that from the early 2000s onwards, although this graph doesn't really continue much into the present, this process has stopped, and in some cases even reversed, because when the temperatures become too high, this is actually bad for photosynthesis. So trees start taking up less and less of the carbon dioxide. Okay, so all these things are possible because of these long-term monitoring plots. So I really encourage the development of a very nice plot network across Indonesia. Would be really informative. Other things that you can do with plot networks are here. This is at a global scale. These are hundreds of plots, and we just mapped the tree diversity. So you can recognize a very diverse area here in the Amazon, some diversity here in Africa, but also that, for example, that this area in Indonesia with Borneo, Sumatra, and the Malay Peninsula is as diverse as this very diverse region here in South America. So really exceptional diversity. And you can also compare using these plots how these forests differ from each other. So we can really see here that the South American forests are really different in species composition than the Asian ones. And we can even see the gradients of difference between the different forest types. So Africa is somewhere in between. Because we are also collecting lots of data from, for example, the oil industry, they are drilling, so they take soil samples, which we can use for pollen, palynology, so we can reconstruct vegetation types in the past. And because our ability to reconstruct past climates also has increased dramatically, we can actually see how the different regions, so this is an example of Southeast Asia, where you see Borneo here, Sumatra here, and Peninsula Malaysia here, how these regions look in the past. So this is during ice age, so the land area looks completely different because Borneo is connected to Southeast Asia and Java and Sumatra and Malaysia because the sea levels were down by about 100 meters. But because we can reconstruct the climate and we also have the pollen data, we can actually see what kind of forest there was where in the region. We can nowadays also do the same thing for the future. So this is a map of Europe, where under a certain scenario of climate change, they have predicted how the species composition will change into the next few decades. And then you can identify regions like these red regions where the species diversity will go down, and other regions like these grayish regions where the species diversity will increase. And they can also predict what kind of species will decrease and what kind of species will increase. So people can already anticipate.
After the loss of potato, sweet potato in the root and tuber crops, but in the fiber crops and the vegetable, this is the main component is mainly from the cellulose, like sugar crop, oil seed crops in oil palm or coconut, beverage crops, tea, coffee. Byproduct is fibers, is the main component. Mass. Actually, there are some treatment we are given to the biomass. Pre-treatment produce reactive cellulose fiber, and also can broken the rigidity of the biomass. Irradiation by using the microwave treatment and also gamma ray treatment can also broken the structure, the crystallinity structure of the lignocellulosic material. Thermal treatment such like autoclaving or high pressure and high temperature treatment is a part also as a physical. And in our research, we have also can eliminate the lignin in the biological pretreatment. And of course, some microorganisms produce enzyme such like ligninase or hemicellulase and cellulase in the from the biological treatment. Some chemicals also will remove the lignin and also the lignin, the hemicellulose from the structure of the biomass in using the alkali, the acid, explosion, gas, oxidizing agent is also with the solvent extraction. So we can choose which one is the pre-treatment is proper with our biomass. It's really depend on the characteristic of the biomass. In detail, we will see the some pretreatment to improve the enzymes of the glucose, but they have different in the linkage between each monomer. Such like in cellulose, they have beta 1,4, but in the starch, they mainly having the alpha 1,4, and also the branching linkage is with the alpha 1,6. So we can see some research about this, and let me see. Let me show you. Okay. In this research, we found that the milling process can reduce the particle size of the sweet sorghum bagasse. In this research, we can see sweet sorghum bagasse was milling and separated into 3 fractions less than 250 micrometers, and the bigger particle more than 420 micrometers, and this is the between the higher and the smaller one. And the cell wall was broken. Maybe this is the effect of the pressing during the juice extraction, and also during the milling process. Sometimes a hammer mill or dish mill give a lot of energy and makes the friction between the particle also will produce energy, heat, and it can also broken the structure of the fiber. And from this table, from this graph, we can see that the smaller particle have more porous, as we can see this one. The higher one is a little bit smooth, yeah, but we can see there are some destruction is made the smaller particle have the more porous and high surface area, so it becomes easy for them to the enzyme susceptibility. The graph shows that the smaller particle with the excess enzyme, this is the cellulose enzyme, can produce higher polysaccharide conversion and also higher reducing sugar yield if we compare to the other particle size. The pretreatment by using the alkaline solution and also hydrothermal. Hydrothermal, it means high pressure and high temperature, also will increase the purity of the cellulose. This picture shows us that the more higher crystallinity, it means it more high level the purity of the cellulose. And by using the alkali hydrothermal, the purity is higher compared to the only the native one. This is the native bagasse. And if we only boiling in the hot water and compared to the also to the boiling alkaline water and only hydrothermal without alkaline solution, so the alkaline hydrothermal give the best purity for the cellulose fraction. This microscopic observation also we can prove that the native, this is the native, is still have the natural structure, but only by using the hot water, maybe the extractive process, the extractive fraction of the extractive component removed, but in alkaline there are some destruction, maybe the lignin also removed, and by using the hydrothermal, a little bit higher temperature, it's a little high pressure, also broken the structure. But using the alkaline, we can see there are a lot of you can see this one, we can see a lot of porous produced after this treatment. So the effect of the alkali and hydrothermal also influence to the enzyme susceptibility. Actually, this treatment was given by using the alkaline hydrothermal, by using the excess enzyme. Also, will produce the higher polysaccharide conversion compared to the other. But with the limited enzyme, maybe not all the structure was converted. So we can say that in the excess enzyme, actually the alkaline hydrothermal give the better the lignification process. This is also similar with the Kumar et al. With his finding, the effect of the alkaline hydrothermal on the biomass porosity and also to the internal surface. This is caused by the high removal of the lignin on partial solubilization of the silane. So I think the process, this is a good combination between the alkaline and also the hydrothermal treatment. Yeah. Okay. This slide show the effect of the microwave treatment as a physical treatment on the enzyme susceptibility of the sago pit. Yeah. Sago pit is the inside part of the sago trunk. This is the main raw material for the sago industry, and the sago trunk consists mainly not only fiber but also starch. Yeah. As we see in this microscopic structure for the native sago, we can see this is the fiber, and this one, the smallest one with the blue and yellow structure, is the starch. Yeah. In the conventional heating, the heat is transferred to the surface of the material and into also the interior of the material by thermal conduction. But microwave, they are not formed of the heat. So the heat is comes from the interaction inside the materials itself. So the heating, it comes from the inside to the outside, but the conventional heating from the outside to the inside of the materials. In the microscopic picture, we can see that the native starch with the high crystallinity in fiber and also the starch. And then after it was gelatinized in the water, and by using the microwave treatment, the starch was gelatinized. But not only the starch, the fiber also swelling, and they change the crystallinity into the more amorphous fraction. So the microwave treatment not only working to the starch, but also to the fiber to make it more susceptible to the enzyme susceptibility. If we compare to the dilute acid solution in the microwave treatment, maybe because the microwave treatment, sometimes with 100% power level, they can reach until 200 degrees Celsius. So there are some further process, especially for the glucose, they can convert it into the furfural or hydroxymetylfurfural. So that's why in this picture, we can see there are a black spot. This is identifying the browning component. Maybe it comes from the furfural or hydroxymetylfurfural. But in the dilute acid treatment, actually, the starch and also the fiber will more excessive heating, and it becomes more easier to the substrate to be hydrolyzed by using the enzyme. Okay. The microwave treatment not only gelatinize the starch, but also swelling the starch and the fiber. And the heat treatment, we compare between the autoclaving 121 degrees Celsius and microwave treatment, and then we combine the enzyme for the liquefaction, because we combine the two enzymes with the almost similar temperature condition. In this research, we find the similar pH and also the temperature for both enzymes working. And then it was followed with the saccharification process, and then we combine also with the amyloglucosidase and also with the cellulose. And after the autoclaving, this is the autoclaving. Actually, the enzyme for the liquefaction is give more susceptible with the microwave treatments products, because they can having more total soluble sugar compared to the autoclaving. But after the saccharification, both of them give almost similar, even some of them slightly increase in the microwave treatment, but they give still higher degree of polymerization of the polymer. So actually both of them having the similar process. But autoclaving takes a very long time. We need 15 minutes to conduct the autoclaving treatment, but using the microwave, we can a shorter time, and this result maybe we find only 30 seconds until 50 seconds is enough to improve or to gelatinize and swelling the starch and also the fiber. This picture shows the macroscopic structure of the sagopit residue after the enzymatic saccharification. We can see this is from the autoclaving. Autoclaving only working to the starch only, but the microwave treatment not only working to the starch but also to the fiber. So we can see the rigid structure of the cellulose was converted into more amorphous. So this is as an action of the microwave treatment. Besides the physical treatment, we can also combine the delignification by using several combination of the pretreatment method. We try to use the corn cobs. Corn cobs, half of the corn cobs carbohydrate is cellulose, and most of them is hemicellulose. So corn cobs actually, this is a good source for the hemicellulose, especially for silane. And now it becomes silane. It becomes an expensive raw material for making the xylitol for the pharmaceutical industry, but the rest or the residue from the separation of the hemicellulose, we can have the cellulose fraction. This is the corn cobs, yeah, with almost one ratio, the same ratio between the cellulose and hemicellulose. The lignified corn cobs, yeah, we remove part of the lignin and also the cellulose fraction, yeah. We eliminate the lignin and also the hemicellulose as much, and the alpha cellulose, it was a modified the cellulose fraction. We modified into the more amorphous fraction. These pictures also you can see, this is the native corn cobs with a natural fiber. This is the structure of the natural fiber, but after the lignification, the fiber it a little bit shrinkle because they lost the integrity by the binding of the lignin. Yeah. After we remove also the hemicellulose and another component, so we have the cellulose fraction with a straight fiber, such like in this picture. And then modification into the alpha cellulose also convert the straight fiber into more gel form. Yeah. The pure cellulose, and then we want to it was inoculated by several cellulolytic bacteria. We can see the change of the fiber before and after the fermentation. Before the fermentation, the cellulose was a straight fiber, and then after the hydrolysis, some crystalline structure of the cellulose was cut off and it becomes more amorphous. So we can see there are some area or not a straight fiber anymore. There are some also we can see in this this picture. We can see a lot of holes, yeah, a lot of holes and pores was formed after the fermentation. Yeah. And we can see that the enzyme produced from the bacterial cellulose also broken and also break the crystalline part into the more amorphous. And the graph, yeah, the graph from this picture, we can see that the cellulose fraction, yeah, cellulose fraction and also the lignified corn cobs. Cellulose fraction is more susceptible
di
to
PPI ya,
the corn cobs.
internetnya lancar.
Now we will see some application of the biotechnology and bioengineering in the process and the product development, especially for the carbohydrate commodity. Okay. First, about the corn flour production. Yeah. Corn flour, it was a traditional corn flour was produced from the corn grits. The corn grit was stepping for several hours, and during the stepping, actually there are some fermentations happens to the stepping process, and then after the fermentation, we draining the rest of the water, and we dry in the low temperature for 24 hours, milling into the corn flours. During the fermentation, this is without any introduction of the microorganism. By using the spontaneous fermentation, we found that a lot of the microorganism was growth, and for 48 hours. At the beginning, lactic acid we cannot find, but we can find only amylolytic bacteria. But after 48 hours, a lot of the lactic acid bacteria, also cellulolytic bacteria, amylolytic, and also the yeast. But in the next research, we want to eliminate the amylolytic because the amylolytic will reduce the yield of the corn flour production, and the yeast also we avoid this this microorganism because it will convert some part of the component into the ethanol or acid. So we use only lactic acid bacteria and also cellulolytic bacteria as indigenous microorganism for as a starter in the next research. Okay. After we isolate the indigenous microorganism, it was identified as lactic acid bacteria and the cellulolytic bacteria, and then we try to compare with the spontaneous fermentation, and also using the only lactic acid bacteria, cellulolytic bacteria, and then combine between the both of the microorganism. And then we found that the fermentation actually can be reduced if we found that the spontaneous fermentation need 48 hours to reach the perfect stepping, but the introduction of the microorganism will reduce from 48 hours to 12 hours. And the result from this showed that the fruit fiber was decreased and the yield also will increase. So this is one application of the microorganism to improve the process and the product process efficiency and also the quality of the products. In the traditional corn flour, sometimes they only having short stepping process. Yeah, sometimes only 3 hours to let the grits absorb the water from the stepping water. But this condition only can have a low yield in the percent of the floor yield. This is less than 60% of the yield of the floor. So we try not only by using the microorganism, but also by using the vegetable enzyme. In this research, they use the papain to improve the process efficiency, especially the enzyme, the cellulosic enzyme can reduce the grit hardness, and also it make it easy to milling the grits to make it as a flour. So when we use the excess enzyme until 1% of the stepping solution, they give almost 100% of the flour. It means almost all the grits can be reduced the size to make it the flour. And this graph also showed that the grit hardness reduced markedly with the addition of the enzyme from the papain. Yeah, if there's no enzyme addition, actually stepping only absorb the water, but also it will swelling the grit, but not as much as the treatment with the papain. In the sour cassava starch, the sour cassava starch actually is not a native Indonesian product. It was in South America, and in South America, it was famous as polvillo azedo. It comes from the spontaneous fermentation of the cassava starch, and it was fermented in a tank. It takes very long time, sometimes more than 1 month to the fermentation, and then it was naturally sun exposure by using the sun drying. It should be sun drying, and then they have the sour cassava starch. In Indonesia, sour cassava starch is useful, especially in the traditional snack or kerupuk, because in small scale industry, they prepare by themselves the cassava starch, and it was a very simple preparation, but it takes time. During the spontaneous fermentation of the cassava starch, we can see that the total acidity of the cassava starch is increased. Yeah, but and also it was followed with the lower pH. If we refer to the standard national Indonesia, maybe this sour cassava starch cannot fulfill the requirement for the SNI, but this is a typical cassava starch. They have a typical also functional properties. So it was different if we want to make it as a standard, because it has total acidity and also lower pH. During the fermentation, lactic acid bacteria also dominate the process, and also the yeast and the fungi, because naturally this microorganism actually we can find in the fermentation of some carbohydrate commodity. Functional properties. So it was different. If we try to introduce the indigenous lactic acid bacteria to the sour cassava starch production. So we isolate and then we culture it to make it more easy to the cassava starch to be converted as a sour cassava starch. Some carbohydrate. Compared to the native, to the spontaneous fermentation, actually the spontaneous fermentation takes around 30 until 50 days. So the introduction of the lactic acid bacteria will reduce the fermentation time until 15 days. But the industry still reluctant to use because it takes time, the 15 days. They want only maybe 2 days, but it's very difficult for us to get the higher acidity and also the lower pH. But the fermentation not only increase the expanding capability and also the swelling power. Swelling power, it means the capability for the starch to absorb the water, and also increase the solubility, digestibility is lower, so it becomes a very good source for the resistant starch. The vascularity also increase, so the introduction of the lactic acid bacteria not only shorten the fermentation time, but also improve the functional properties. Some traditional implementation of the cassava production is a small scale industry. So sometimes they give or use the river water as water for the production. So a lot of the pathogenic microorganism from the supply water. So the introduction lactic acid bacteria also can improve the food safety because they can inhibit the pathogenic bacteria such like as Escherichia coli or Salmonella. This picture shows the microscopic from the light microscopic. This is a 0 days. There are no knot or porous in the center of the granule. But if we see the effect of the fermentation from the starch granule, a lot of spot or some hole inside the granule. It means that the lactic acid bacteria produce a lot of acid, and the acid also will make some erosion on the surface of the granule. And beside the lactic acid, some active compound also produced by lactic acid bacteria. It becomes a biopreservative for the products, so we can enhance the shelf life of the product. So it will a very good benefit for them to improve the quality of the cassava starch. This product shows that the increasing of the baking expansion capacity actually can be happen not only to the spontaneous fermentation, but also to the lactic acid bacteria fermentation. But we can see after 50 days, there are only slightly increasing in the baking expansion capability. But using the introduction of the lactic acid, the volume increased markedly. As we can see, this is in the picture. Maybe this is an almost famous product in Indonesia. They like to try such like a cireng, product of the cireng. So we try to measure the volume of the baking expansion, and for the 15 days, they have a very large expansion capability. Also, they have a more lower viscosity to make it more easy to be consumed or chewing the product. It was a very interesting because some traditional industry, they try to make their own product, even a little bit sour smell, but it was have a good functional properties, so they still maintain the process. They cannot use the chemical preservatives such like antimicrobial, because if there's some chemicals, so there's no microorganism will grow in the spontaneous fermentation. So it makes us can assume that the spontaneous fermentation actually it was also an organic process. Even a little bit sour smell. In cacao bean fermentation, there are a lot of microorganisms take part in the formation of the flavor or aroma for the cacao. And in the cacao fermentation, this is from the spontaneous fermentation. The fermentation takes several days, and lactic acid will dominate the microorganism in the beginning or in the initial process. And the acetic acid maybe takes more than 3 days after the fermentation, but yeast also dominate in the initial process. A lot of product was produced during the fermentation. And there are some conversion from the fructose and glucose. The fructose and glucose, it comes from the pulp of the cacao bean. Yeah. So the fructose and glucose was converted into some intermediate products such like ethanol, acetic acid, lactic acid, also conversion of the fructose into the mannitol. All of the compound is important in the fermentation of the cacao bean, since it was contribute to the flavor formation. Okay, we can see some research. There are some. This is the some standard for the cacao bean quality. In the farmer, cacao bean fermentation conduct using the spontaneous fermentation, and this is fermentation produce low quality of the bean, and it was give lower price with low flavor and aroma of the cocoa product. So it was giving a very low added value to the farmer. So the fermentation, spontaneous fermentation, we try to compare with the lactic acid bacteria, the introduction to the lactic acid bacteria. Yeast, also the combination with between the lactic acid bacteria and the yeast. From the spontaneous fermentation, the fermented bean is very low. But if we introduce the lactic acid bacteria, the fermented bean was increased, and the combination gave the best result until 83% of the fermented bean has been produced from this fermentation. But unfermented also will reduce. Non-fermented, it means it's still a raw cacao. Germinated, yeah, actually germinated cocoa bean. It was a poor quality because some of the component was converted into the new generation of the plant. So the germinated was they germinated was the part of the ethanol to inhibit the germination of the cacao bean. This is the slutty. It means give the bad flavor and aroma. We have to reduce as much as we can. So from the best fermentation, a lot of fermented we can produce, but we can also reduce the non-fermented germinated on the side, slutty coffee bean. As we can see, this is from the bean that we can produce from the fermentation. Yeah, this is a very good fermented, produce a dark, sometimes a purple color. Yeah, but it is non-fermented. It's still a green one. So this is a germinated. A lot of germs comes here. And also slutty, it becomes rotten one. The pH also become the alkaline. Actually, the cacao bean process, the alkaline process is a very important because they have a very good flavor and aroma if we want to use in the industry. This is a very good fermented, produce a dark, sometimes a purple color. In the cafe, yeah, in the cafe, we also introduced some microorganism, actually from the Actinomycetes. as a starter for the coffee fruits fermentation, because we want to take some active compound from the coffee pulp. Actually, the coffee cherries, yeah, there are pulp and also the bean. And in the wet fermentation, they fermented the whole cherries. We did not separate the coffee pulp, but after the fermentation, we have to separate between the coffee pulp and also the coffee bean, so we can take the active compound from the coffee pulp to take some active compounds such like as a polyphenol, tannin, anthocyanin are also catechin. But we can see that the polyphenol was increased by the fermentation, but it can also reduce the bitter components such like tannin, increasing the anthocyanin antioxidant activity, also the catechin as a part of the flavor in the coffee. So the introduction of the coffee, the microorganism, they can improve the quality of the coffee, green coffee, but also we can take some valuable product, especially the bioactive compound from the coffee pulp. Dietary fiber production from the cassava pulp also can be used. We can use some cellulosic microorganism. This is the cassava pulp. Cassava pulp actually from the small scale industry, they have two main component. It has a starch and also fiber. Yeah. So the fiber, we can make the fiber to make the dietary fiber. Cassava pulp in a not inefficient process, they contain a lot of starch. So sometimes we have to eliminate, but we can take the starch component by using the enzyme. So we will have the glucose syrup. And then the residue is a cassava pulp fiber. We give the enzymatic treatment to produce a dietary fiber. Such like in this picture, the cassava pulp sometimes was dried, and it still can be used as the food component because it still have a lot of fiber and starch. This picture shows the scanning electron microscopic of the cassava pulp fiber before the hydrolysis. There are two treatments we can give, the thermal process and the non-thermal process. Non-thermal process, it means only boiling in the water, but thermal process, we use the autoclaving process. So this is from the cassava pulp fiber before the treatment, the enzymatic, from non-thermal and also to the thermal process. There are some destruction in the fiber we can find with the high thermal process, but after the hydrolysis by using the cellulose enzyme, a lot of the destruction will be exposed with the thermal treatment. So we can find there are a lot of degree of the value is reduced during the fermentation time. And also if we compare between the thermal treatment and the non-thermal, actually it's not so different. But if we find the functional properties, this is the soluble fiber and also insoluble fiber, they are not so different between the composition, but they can say that the functional properties, its water holding capacity, oil holding capacity, and also emulsifying capacity increase with the thermal treatment and the fermentation time. So the cellulolytic enzyme and also the thermal treatment will improve the dietary fiber properties and also the number of the soluble dietary fiber. Different between the composition. As a conclusion, we can say that the biotechnology or bioengineering have important roles in the carbohydrate bioconversion, especially in improving the efficiency of the process, to improve the quality of the product, to produce valuable product, and to add the value of the agriculture materials, and to conduct the clean production, to eliminate the waste and the byproduct in agro-industry. Okay, I would like to all the data presented in this lecture was produced from all my students' publication and also from my colleague. I would like to thank to all of them to support me in this presentation. Thank you very much. And terima kasih. The value of the agriculture materials and to conduct the clean production, to eliminate the waste and the byproduct in agro-industry. Okay, I would like to all the data presented in this lecture was produced from all my students' publication and also from my colleague. I would like to thank to all of them to support me in this presentation. Thank you very much. And terima kasih. The value of the agriculture materials and to conduct the clean production, to eliminate the waste and the byproduct in agro-industry. Okay, I would like to all the data presented in this lecture.
Thank you very much for Ibu Titi for the very brief presentations. We are really sorry because of the connection trouble. We're still waiting for Ibu Eis to join us again. Ibu Eis, are you ready?
Yeah, okay, ready. I'm sorry for the technical problem. Thank you very much to Dr. Titi for the interesting presentations. Okay, for all
Thank you very
participants,
much
we
for
have
Ibu Titi
already
for the very
listened to the
brief
presentation
presentations.
from Professor
We are really
Slick
sorry
and Dr.
because of
Titi.
the connection
Professor Slick has
trouble.
already
We're
explained that
still
we
waiting
are
for
in
Ibu
the
Eis to join
situation of
us again.
environmental
Ibu Eis, are you ready?
crisis that affects biodiversity in Southeast Asia and other parts of the world also. However, on the other side, there are some new scientific developments that could help us to better understand, study, and monitor biodiversity and its changes. And Dr. Titi has explained about carbohydrate resources that are abundantly available and the role of biotechnology and bioengineering in the conversion and transformation of these carbohydrates into different kinds of useful and value-added products. Now, I would like to invite questions or comments from the audience. I have seen one question from Dani Satyawan from ICA Biograd, is that right? To Professor Ferislik. His question is, what are the 3 DNA barcodes you mentioned that are needed to identify plant species with high certainty? Also, how good is the ability of nature to reclaim areas that we completely abandon? Is there any other questions? You may raise hands or his question is, what are the 3 DNA barcodes you mentioned that are needed to identify? Okay, Dr. Slick, please just start answering.
I still have a problem with my camera. I'm sorry for that. The 3 regions for identifying plants, though it's not really my specialty, but I think it's RBCL, MATE, and what was the other one? ITS, and sometimes they had even a fourth one. So each of those, they can actually they cover different levels of variation in the DNA. So the RBCL is very good in identifying family level, for example. And the regions for identifying or plants, what was the other TRNL, really my specialty, and especially the ITS, they are good for identifying genus, species, and sometimes even within species level differences. And sometimes they had even so it depends a bit on how detailed you want to identify your plants, but you have to keep in mind that there are several groups of plants, for example orchids or gingers, that even if you use those 4 regions, often you cannot still not identify all the way to species. So it's like a tool that you can use for most species, but not always all species. So that's a bit problematic for plants. For animals, it actually works much better, and for fungi. It depends a bit on how detailed. And the other one for the, you have to keep in mind that the restoration of areas, yeah, it's actually really good if you can follow the natural succession. Often, if the disturbance has not been too severe, or there is still natural forest nearby, you can often rely on natural regeneration. It's often even better than if you start interfering yourself, because when people start doing reforestation, they often don't get the species right, or the abundances of the different species within the new system. Natural regeneration, they have developed some good systems in Thailand, I think, where they have a set of standard species that they use, that especially species with fruits that can attract seed dispersers like birds or mammals. So they start out with those few species, and once they start flowering and fruiting, they will attract these natural seed dispersers. And other species will be brought into the system that you started with these basic, they call them framework species, I think. So if possible, I would always prefer natural regeneration. It's also much cheaper. But sometimes that's not possible. If there is no more natural forest nearby, you really have to start helping the regeneration. Okay, I hope that answered the question.
Okay, is that okay to Mr. Dani Satiawan? No? Yeah.
Very clear.
Pak Dani, is that okay?
Yeah, very clear. Thank you very much.
Okay, thank you, Pak Dani. Thank you, Professor Slick. There is another question from Dr. Ruliana Susanti from LIPI. What is the main challenges that you face when developing the plot network? In the case of Indonesia, we have so many permanent or semi-permanent plots in smaller size, less than 1 hectare. I was wondering how to connect all those plots when the methodology used is different. Please share your experiences. Please, I think you may directly answer the question.
One of the biggest problems that I always face when I do plot analysis, they all differ in shape and size, which can really influence the analysis, because if you compare like a square plot with a transect, you have a different level of beta diversity in your plot. So actually you could not directly compare them. So I cannot really exclude these problems, but what I normally do is I use, for these global analyses, I used like 1 degree grid cells. So that's for the at the global scale, 1 degree latitude, 1 degree longitude. And I just put all the plots together that are found within that line. Be biased because of that, but that still leaves quite a lot of variation that you can explain using this kind of method. So there is no perfect solution for this. You can just try to minimize the problems. So plots all over Indonesia, even though they are all different sizes and different shapes, you can still do a very nice analysis. Be biased because of that. My camera.
Have you completed the to answer? Okay. How is it, Dr. Ruli?
Be biased
is
because
that answer your question?
But that still leaves quite a lot of variation.
Okay, thank you, Professor Slick. The next question is to Dr. Titi Chandra Sunarti from Sylvia Kurniasari, Universitas Erlangga. Thank you for the presentation. My question is, why the production of corn flour in this study performed by living cells instead of using crude enzyme, since the product can be utilized by micro to reduce the product? Thank you. Please, Dr. Titi.
Yes, thank you to Sylvia. That the microorganism will reduce the yield of that in the spontaneous and the process. They use the stepping process, and during the stepping process, a lot of microorganisms also grow in the fermentation, and it takes a longer time. So if we introduce some microorganism, so it will shorten the fermentation time, so we can save. Not all the component will solute into the water of the stepping process. So even the microorganism will consume some of the component in the stepping water, but shorten. Not all the microorganism can consume the part of the component inside the grids.
Is that okay, Ibu Sylvia? Okay, thank you. The next is from Izar Khairullah Isari to Dr. Titi. Is there any difference between carbohydrate from roots, crops, and from fruit plants, such as from rice?
Okay.
Please, Dr. Titi.
Yes. For carbohydrate form in the fruits and only in the roots, of course, it was totally different. Such like in the roots, in the cassava, and also in the sweet potato, yeah, they have a lot of fiber and starch. But in the fruits, such like ordinary fruits, they a lot of simple sugar and also fiber. But such like wheat, because it was a cereal only starch, but also contains some lipid and protein. It becomes different in how to handling this commodity. So between the part of the plant make it different in the carbohydrate composition.
Thank you, Bu Titi. Dr. Pak Izhar, bagaimana? How is the answer? Is that okay with you?
Okay, thank you for your explanation, Dr. Titi.
Thank you to Dr. Titi. Is there any other question from the audience? We still have about 15 minutes for discussion sessions. While waiting, another question from the audience. Probably I would like to ask a question to Professor Slick. This is just general questions because I'm not an expert in biodiversity. Do you think Southeast Asia countries have enough efforts to prevent biodiversity loss or to manage biodiversity crisis? And how is it compared to other countries in Asia, for example, or other part of the world?
That's a very difficult question.
Oh,
I think,
I'm sorry.
yeah, all the countries they differ in governments, and but I think overall most of the countries have the capacity to do it, and probably also they have a will to do it. The problem is mostly that it is they have to make a compromise between development and conservation. So the thing is to try to mix the two, where you can both develop and conserve. So that needs a good amount of planning, and also very good and tough law enforcement, which can be very difficult because it differs per country, right? How you can implement that. So I think it's very well possible. There's also already a lot of international collaboration within ASEAN, and we have these big projects like Heart of Borneo, and there was this, I think it's still running, the Greater Mekong project in the Asian mainland. So I think the will is there, and actually I'm still quite positive about the future. Although there are still things going wrong, I think we still have the ability to turn it around.
Okay, thank you very much. And there is still no question from the audience. Probably one more question to Dr. Titi. This is also general questions. There are different kinds of new products, for example, bioethanol for energy and different kinds of polymers for packaging that can be produced from carbohydrates, including starch. Do you think there will be enough starch for food? Because most people, especially people in developing countries, use starch as staple food. What is your suggestion regarding this matter?
There are some competition between the food and energy, especially for the carbohydrate commodity. But we have to find another source for the carbohydrate for the non-food application, because in the global market, the starch source comes from the cassava, especially in the tropical countries. But in the subtropical countries, they use corn and also potato. So in the future, maybe we have to convert the bioenergy not comes from the simple carbohydrates, such like from the sugar or from the starch. So we can go to the next generation of the bioethanol production. Yeah, we can use the residue from the agro-industry, such like solid waste, which can contain a lot of rich in cellulose and also rich in starts to become the source for the bioethanol production. But don't forget that we have unexplored carbohydrate source, such like sago. It's still plenty of the potential sago in Indonesia also unexplored. So we can use sago not only for food but also for the non-food application, especially for bioenergy and also for packaging application. Thank you.
Thank you very much, Dr. Titi. Okay, there is some questions from the audience. First one from Ibu Yuni Rahayu from LIPI to Professor Slick. Do you have a suggestion regarding the planning to move the capital city of Indonesia to East Kalimantan from a conservation and biodiversity perspective? Probably I will read the next question also. Maybe Professor Slick could answer it both together. From Pak Sutomo, how does invasive alien species should be handled in terms of ecosystem health? I think, yeah. Yeah. Okay, Professor Slick, please.
Capital. That's kind of a political
topic. Yeah.
I don't know what to think. It seems overall to move a whole capital is a very difficult thing to do. There is one example already in the world, like now several. Malaysia has tried to set up a new capital city, and there is in Brazil they set up Brasilia as capital city. I don't think it's going to be very easy to do this, because you have to move all the government agencies there and the people have to follow, and it's a huge infrastructure project. So for the environment in East Kalimantan, it may be a bit risky, but for the economy of Kalimantan, it would be great, of course. So yeah, I don't know exactly. It has to be done in a very, if it's going to happen, it has to be done in a very careful way. It's also a chance, perhaps, if it's going to happen anyway, to show that such a very big move can be done in an environmentally friendly way, right? So there are some opportunities and some dangers, so they have to be considered carefully. For the invasive species, generally it depends a bit on the invasive species itself. Sorry for my camera. Some invasive species are not actually that harmful, so they
Aduh, mati. Oh, I'm sorry. Professor Slick, we cannot hear you.
How much did you not hear?
Okay.
My whole answer, not, or just the last part?
The last part only. The last part only.
Oh, okay. Yeah, so there's different kinds of invasive species, and they will need different kinds of management. So the not so dangerous ones are just an addition to the species that are already there. But the dangerous ones, you either have to try to eradicate them from very early on. So then you need a very good monitoring system to detect them. Or you have to create situations, because we have a big problem in Brunei actually with an invasive species, which is Acacia mangium, which is also in Indonesia.
Yeah.
It spreads everywhere where there is enough light. So, and there are now so many, and especially our heath forests are threatened by this species, and they also cause fire. The only way to get rid of them, because it's now almost impossible to remove them by hand, or it's just too expensive and too much work, is to prevent these fires from happening, so that the natural forest can restore itself and outshade these invasive species, because many invasive species are like disturbance species that need a lot of sunlight. So if you can get some kind of canopy cover back, you might also suppress many of these invasive species. So that I think is the most suitable approach for invasive species that have already established and are really degrading the landscape.
Okay, thank you, Professor Slick. How is Ibu Yuni and Pak Sutomo? Is that okay?
It's okay, clear. It's thank you. For
Okay, there is one more question. I think this is the last question because the time is almost up. From Ibu Sylvia Kurniasari, Universitas Erlangga, also to Dr. Titi. My question is, the use of papain enzyme was to reduce the hardness of the substrate. Is it possible if we directly add plant extract that proven contain papain enzyme into the fermentation? And how papain enzyme can reduce the substrate hardness? Thank you. I think this is the last question. Okay.
Okay, thank you, Ibu Sylvia. Actually, if we directly milling the grits to make flour, only around 30% of the flour that we can produce. But addition of the cellulosic enzyme, such as papain in the research, so it will break the hard endosperm, which the protein matrix. So that's why we add the cellulolytic and also proteolytic bacteria. So that's why the hard protein matrix should be break, and it becomes easy for them to milling the hard endosperm of the corn grits.
Okay, how is it, Ibu Sylvia? Is that okay?
it's okay, thank you.
Thank you, Ibu Sylvia. Okay, then I think this is the end of the first plenary sessions. I would like to thank again to Professor Ferry Slick and to Dr. Titi Chandra Sunarti, as well as to all the participants for joining these sessions. I hope you enjoyed these sessions, and I give back the chair to Ms. Eva. Ms. Eva, please.
Thank you very much, Professor Ais and the speakers for the informative session. Before proceeding to the next session, I would like to invite all speakers, distinguished guests, and participants to join a photo session. You are pleased to activate your camera. Please be ready.
Okay.
Okay.
Thank you very much to all participants. I would like to remind you to fill the list of attendees via the link provided in the chat. And also, please be aware to assign your username as indicated by the conference rule to enable us to organize you according to your room. For the oral participants, please assign your username using code of room _ code of presenter _ name. And for non-oral participants, you also need to change your username using code of room _ participant _ name. Our committee will organize you according to your room. Ladies and gentlemen, we are now having a break session. We will start the parallel session at 1 p.m. We will have a parallel session in 2 rooms. Room 1 will be hosted by Dr. Renat Ayu Safitri Nurinsyah, and room 2 will be hosted by Dr. Cahyo Rahmadi. At the end of this symposium, we will announce the best presenter from each room. Please join on time. The participants may either stay online or leave the meeting. However, for those who leave the meeting, we require you to join again the symposium at 12:30 using the Zoom link provided by the committee. Thank you and see you in the next session.
Every vision begins with a spark of inspiration, and every inspiration is driven by the passion to becoming a world-class institution in research, sciences, and innovation to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, LIPI. LIPI has various research centers throughout Indonesia under the coordination of the chairman of LIPI, Deputy for Life Sciences, Deputy for Earth Sciences, Deputy for Engineering Sciences, Deputy for Social and Humanities Sciences, and Deputy for Scientific Services. LIPI has a pivotal role in drafting and establishing government regulations and policies related to economic, political, social, and cultural aspects. LIPI also manages scientific data and scientific considerations on biodiversity conservation, as well as the publication of global indexed scientific journals, and also appointed as a scientific authority in coaching national researchers and hosting the inauguration of Indonesian research professors. To support world-class research activities, LIPI managed 5 botanic gardens as a center for ex situ plant conservation, 3 museums that have the biggest collection of flora and fauna specimens in Southeast Asia and regional level and Indonesian natural history, Barunajaya 8 research vessel, Indonesia Cultural Collection, which keeps the largest collection of microorganisms in Indonesia, and 2 international centers, namely Asia Pacific Center for Ecohydrology, APCE, and Regional Training and Research Center on Marine Biodiversity and Ecosystem Health, RTRC Marbest Center. LIPI has produced qualified research results that are beneficial to the society, including biodiversity, environment and maritime, food security, agriculture and farm, health and pharmacy, advanced material, energy, technology, information and communication, social dynamics, humanity and culture, technology innovation and utilization of science and technology, as well as scientific services, testing and development. LIPI's national scientific collaborations include collaborative research, technology licensing, development, application, and commercialization of research results. Non-research collaborations, LIPI's expertise consulting, scientific coaching for youth, students, and teachers with various universities, research institutions, local governments, and industry. As for international collaborations, LIPI conducts joint research, joint laboratories, symposium, workshops, international conferences, exchanges for researchers and experts, as well as international scholarships, while being active as a member and a focal point in various international organizations. LIPI will keep continuing to strengthen national platforms by developing world-class researchers and building scientific collaborations to make great impacts on the nation's competitiveness.
Secara prinsip etnobotani itu dibangun dari dua disiplin keilmuan, yaitu ilmu antropologi dan ilmu botani. Jadi gabungan dari dua disiplin ilmu ini melahirkan atau satu disiplin ilmu, yaitu etnobotani. Kaitannya sangat erat, yaitu bagaimana memahami relasi orang dengan alam sekitarnya. Jadi relasi orang dengan tumbuhan itu seperti apa? Misalnya bagaimana orang melihat dan memanfaatkan tumbuhan untuk obat-obatan, pangan, energi, kosmetik, kesehatan, dan macam-macam. Artinya itu hubungan orang dengan tumbuhan. Dalam hal ini biasanya pemanfaatan. Nama saya Muhammad Fatsir Royani, saya peneliti etnobotani, bidang botani, Pusat Penelitian Biologi, Lembaga Ilmu Pengetahuan Indonesia. Etnobotani oleh beberapa kalangan itu dianggap sebagai satu disiplin ilmu sendiri. Artinya disiplin ilmu itu dia memiliki metode. Tren sekarang sebenarnya etnobotani itu tidak hanya ke arah penggalian informasi semua jenis tumbuhan yang dimanfaatkan oleh masyarakat. Sekarang orang bisa melihat melalui satu spesies tumbuhan tertentu, kemudian didalami betul dari A sampai Z-nya, dari pemanfaatan misalnya dari pengolahan, pemanfaatan sampai kemudian pemanfaatan akhir atau misalnya keuntungan ekonomi dan lain sebagainya. Karena background saya sebenarnya awalnya saya adalah sejarah, dan sejarah itu mau tidak mau dekat dengan antropologi. Bahkan ada adagium, sejarah adalah antropologi masa lalu dan antropologi adalah sejarah sekarang. Nah, artinya dekat. Kemudian saya melanjutkan sekolah dan ambil jurusan antropologi karena memang tadi tertarik saja mempelajari di alam pikiran orang-orang. Saya melihat bahwa kadang kita di balik kesederhanaan itu ada keistimewaan. Di balik kesederhanaan ada kemewahan. Di balik kesederhanaan ada keluar biasaan. Nah, itu saya tertarik. Kemudian masyarakat-masyarakat yang tinggal dekat dengan hutan, jauh dari perkotaan segala macam, itu diasumsikan mereka memiliki relasi dengan alam yang sangat baik. Serta tumbuhan itu tiap hari berelasi dengan manusia, sehingga asumsi saya karena selalu terjadi relasi antara manusia dengan alam, maka pemahaman manusia itu terhadap alam, terhadap tumbuhan itu sangat baik. Itu kenapa pilihan lokasi lebih banyak ke masyarakat yang ada di pedalaman atau dekat hutan. Kalau ke depan, saya pikir juga etnobotani harus lebih maju dalam pengertian bagaimana kita tidak hanya memahami secara mendalam perilaku atau pikiran masyarakat, tetapi juga memahami tumbuhan secara lebih holistik, lebih utuh. Misalnya, sekarang kan problem-problem kemanusiaan luar biasa ya. Ada kesehatan, ada kerusakan lingkungan, tapi ya tentunya pintu masuknya adalah informasi dari masyarakat. Kemudian setelah itu kita harus mulai bekerja sama atau membangun jaringan dengan orang-orang kimia bahan alam atau apa untuk mengetahui kandungan-kandungan tumbuhan yang tumbuhan tersebut berdasarkan informasi masyarakat diketahui sebagai obat dari suatu penyakit tertentu.
Pulau Enggano, pulau terluar berpenduduk yang secara administratif terletak di Kecamatan Enggano, Kabupaten Bengkulu Utara, Provinsi Bengkulu. Pulau Enggano memiliki panjang 35 km dan lebar 16 km, dengan luas 402,6 km persegi, dengan garis pantai sepanjang 112 km, dengan titik tertinggi di Bukit Koho Bua-Bua, yakni 281 meter di atas permukaan laut, sehingga dapat dikategorikan sebagai pulau kecil. Perpaduan antara pulau terdepan dan pulau kecil membuat Pulau Enggano menjadi sangat penting terkait dengan program pemerintah menyangkut perlindungan dan penyelamatan pulau-pulau kecil dan terdepan negara kesatuan Republik Indonesia. Oceanic Island menjadi keunikan dari Pulau Enggano. Dengan kata lain, Pulau Enggano dalam sejarah geologinya tidak pernah bergabung dengan daratan besar Pulau Sumatera. Kecamatan Enggano terdiri dari 6 desa yang letaknya di pinggir pantai dan berbatasan langsung dengan Samudra Hindia. Keenam desa tersebut adalah Desa Banjarsari, Desa Meok, Desa Apoho, Desa Malakoni, Desa Kaana, dan Desa Kahyapu dengan pusat pemerintahan di Desa Apoho. Dari segi sejarah, Pulau Enggano pernah menjadi salah satu tempat transit jalur perdagangan internasional pada masa awal kolonialisasi di Nusantara. Hal ini menyebabkan Pulau Enggano memiliki keunikan tersendiri dari segi bahasa, tradisi, dan budayanya. Masyarakat Pulau Enggano terdiri dari 6 suku, yaitu suku Kauno, suku Kaarubi, suku Kaitora, suku Kaharuba, suku Kahua, dan suku Kamai. Mereka menganut sistem matrilineal atau mengikuti garis ibu dalam hal keturunan dan suku. Nama Enggano pertama kali dituliskan oleh Cornelis de Houtman pada saat melakukan ekspedisinya bersama 4 kapal ekspedisi yang bernama Mauritius, Hollandia, Amsterdam, dan Duiven. Pada catatannya, Houtman menuliskan 1596 tiba di Pulau Hindia yang pertama, Pulau Enggano, di sebelah barat Pulau Sumatera bagian selatan. Pada tahun 1771, Charles Miller berhasil berlabuh dan bertemu penduduk asli Pulau Enggano. 100 tahun kemudian, tepatnya di tahun 1865, Mr. Francis datang ke Pulau Enggano dan mendapatkan kesan yang mendalam dari penduduk Pulau Enggano. Hal tersebut diketahui oleh seorang linguistik asal Jerman, Hans Kehler, yang mengunjungi Pulau Enggano pada tahun 1930-an. Adalah kemudian seorang pengelana dunia asal Italia yang bernama Elio Modigliani, yang kemudian mencatatkan Enggano dalam sejarah melalui bukunya L'Isola delle Donne, Viaggio ad Enggano. Elio Modigliani berlabuh di Pulau Enggano pada tahun 1891 dan tinggal di sana selama 8 bulan, yang kemudian mencatatkan temuan keanekaragaman fauna di Pulau Enggano. Di tahun 1936, naturalis asal Belanda, Wilhelm Jan Lujham dan Jocelyn de Jong melakukan ekspedisi dan koleksi flora dan fauna di Pulau Enggano. Dan di tahun 2015, Kedeputian Ilmu Pengetahuan Hayati, Lembaga Ilmu Pengetahuan Indonesia, melakukan ekspedisi Widya Nusantara untuk meneliti keanekaragaman hayati, menemukan potensi bioaktif dari taksa terpilih yang dapat dikembangkan sebagai bahan pangan, obat-obatan, dan sumber energi. Kegiatan penelitian ini dirancang bersinergi dengan berbagai aspek, mulai dari eksplorasi, identifikasi, hingga analisis potensi dan pemanfaatan.
Enggano dipilih sebagai prioritas. Jadi kalau di Kedeputian Ilmu Pengetahuan Hayati LIPI itu ada beberapa program unggulan LIPI, dan salah satunya adalah eksplorasi bioresources di Pulau Enggano. Jadi ini merupakan salah satu kegiatan unggulan Kedeputian Ilmu Pengetahuan Hayati. Kenapa dipilih Enggano? Itu ada beberapa pertimbangan. Pertama adalah pertimbangan saintifik, itu karena pulau ini adalah oceanic island, di mana pulau ini dalam sejarah geologinya itu tidak pernah bergabung dengan Pulau Sumatera. Nah, konsekuensinya itu jenis-jenis atau spesiasi yang ada di pulau ini tetap unik, banyak jenis-jenis endemik, dan yang paling mendasar adalah informasi tentang jenis-jenis itu belum banyak. Jadi belum ada data-data yang lengkap mengenai jenis-jenis yang ada di pulau tersebut. Yang kedua, secara geopolitik, pulau ini merupakan salah satu pulau terluar di kawasan Nusantara, di mana pulau-pulau kawasan terluar itu menjadi prioritas pembangunan, sehingga untuk melaksanakan pembangunan itu harus ada data-data yang mendukung. Data-data yang mendukung adalah data dasar potensi dari pulau tersebut, termasuk adalah eksplorasi potensi bioresources Pulau Enggano.
Kegiatan ekspedisi Pulau Enggano dilakukan oleh para peneliti dari Pusat Penelitian Biologi, Pusat Penelitian Bioteknologi, Pusat Konservasi Tumbuhan Kebun Raya Indonesia, dan Pusat Penelitian Biomaterial. Kegiatan ekspedisi bertujuan mendata jenis-jenis flora, fauna, dan mikroorganisme yang ada di Pulau Enggano, salah satu pulau terdepan di Provinsi Bengkulu. Dengan keunikan sejarah geologinya, Pulau Enggano diyakini memiliki tingkat endemisitas flora dan fauna yang tinggi. Pulau Enggano memiliki ekosistem khas pulau kecil, ekosistem alami seperti ekosistem laut, ekosistem semi-terestrial seperti mangrove dan tepi sungai, serta ekosistem terestrial, yakni hutan pantai, hutan dataran rendah, dan rawa. Ekosistem hutan pantai dapat ditemukan hampir di sepanjang garis pantai timur Pulau Pulau Enggano. Karakteristik pantai yang terdapat di Pulau Enggano adalah pasir berlumpur, pasir berkarang, pasir karang berlumpur, dan pantai karang berbatu yang keberadaannya berkaitan erat dengan ekosistem terumbu karang dan mangrove. Peneliti dari Pusat Penelitian Bioteknologi dan Pusat Penelitian Biologi mengeksplorasi dan mengumpulkan mikroorganisme dari air laut, sedimen laut, pasir laut, spons, tanah, serasah, daun, bunga, buah, jamur endofitik, dan jaringan atau organ hewan. Koleksi mikroorganisme ini dilakukan untuk mencari agen pembentukan senyawa obat, penghasil energi, dan penghasil enzim penting dalam sektor pangan dan pengolahan produk pangan. Dalam ekspedisi ini, para peneliti telah berhasil mengkoleksi mikroorganisme antara lain antinobakteria, bakteria, khamir, jamur, arkea, dan alga. Indonesia telah dikaruniai ragam hutan alam yang terbentang dari ujung barat di Sabang sampai ujung timur di Merauke, yang merupakan habitat utama berbagai jenis flora dan fauna. Hutan pantai merupakan bagian dari ekosistem pesisir dan laut, menyediakan sumber daya alam yang produktif, baik sebagai sumber pangan, penghasil obatan yang bernilai ekonomi tinggi, penemuan produk kimia, tambang mineral, dan kawasan wisata pantai. Pulau Enggano memiliki hutan mangrove paling luas di Provinsi Bengkulu, yakni 1.710 hektar. Tanjung Kaana merupakan daerah yang mempunyai hutan mangrove paling lebat. Selain hutan mangrove, Pulau Enggano juga memiliki daerah rawa dengan luas 1.967 hektar. Pada kedua ekosistem ini, ditemukan mikroorganisme yang dapat hidup dalam kondisi anaerob atau kondisi tanpa oksigen. Ekosistem mangrove banyak ditemukan di Pulau Enggano. Jenis dominan seperti Bruguera gymnorrhiza yang dapat ditemukan di sisi tenggara dan barat daya pulau. Hutan mangrove yang tumbuh di pantai daerah tropis merupakan habitat bagi berbagai hewan. Burung, mamalia, dan reptil mendapatkan keuntungan dari kayanya pasokan makanan di wilayah ini seperti ikan, kerang, tiram, siput, anemon laut, dan kepiting. Selain itu, hutan mangrove melindungi pantai dari terjangan gelombang dan banjir. Di Pulau Enggano mengalir beberapa sungai besar dan kecil yang memiliki hulu di bagian tengah pulau. Kita dapat menemukan jenis tumbuhan seperti Nipa frutican atau nipah, Sakarum atau gelagah, Pometia pinnata atau matoa, serta pada daerah sungai yang dekat dengan tepi laut dapat ditemukan Terminalia catapa atau ketapang, dan Barringtonia macrocarpa atau putat. Di Pulau Enggano, ditemukan jenis tumbuhan berkayu potensial yang telah dimanfaatkan dan memiliki potensi ekonomi seperti merbau, Hutan dataran rendah yang masih merupakan hutan primer masih dapat ditemui di bagian tengah pulau. Sedangkan hutan dataran rendah yang digolongkan sebagai hutan sekunder banyak ditemukan pada bagian utara pulau, di mana pengaruh antropogenik paling banyak ditemukan. Banyak tumbuhan berkhasiat obat yang tumbuh di Pulau Enggano. Selain itu ada pula komoditas pertanian yang utama di Pulau Enggano, yakni budidaya pisang, coklat, kedondong, dan salak yang tumbuh liar di hutan. Beberapa komoditas pangan yang belum maksimal terungkap di Pulau Enggano adalah buah pandan dan buah nipah. Tumbuhan paku banyak ditemui di Pulau Enggano. Selain itu, suku talas-talasan dan anggrek juga menjadi jenis kekayaan hayati yang akan diaklimatisasi dan ditanam sebagai koleksi kebun raya Indonesia.
Secara prinsip etnobotani itu dibangun dari dua disiplin keilmuan, yaitu ilmu antropologi dan ilmu botani. Jadi gabungan dari dua disiplin ilmu ini melahirkan atau satu disiplin ilmu, yaitu etnobotani. Kaitannya sangat erat, yaitu bagaimana memahami relasi orang dengan alam sekitarnya. Jadi relasi orang dengan tumbuhan itu seperti apa? Misalnya bagaimana orang melihat dan memanfaatkan tumbuhan untuk obat-obatan, pangan, energi, kosmetik, kesehatan, dan macam-macam. Artinya itu hubungan orang dengan tumbuhan. Dalam hal ini biasanya pemanfaatan. Nama saya Muhammad Fatsir Royani, saya peneliti etnobotani, bidang botani, Pusat Penelitian Biologi, Lembaga Ilmu Pengetahuan Indonesia. Etnobotani oleh beberapa kalangan itu dianggap sebagai satu disiplin ilmu sendiri. Artinya disiplin ilmu itu dia memiliki metode. Tren sekarang sebenarnya etnobotani itu tidak hanya ke arah penggalian informasi semua jenis tumbuhan yang dimanfaatkan oleh masyarakat. Sekarang orang bisa melihat melalui satu spesies tumbuhan tertentu, kemudian didalami betul dari A sampai Z-nya, dari pemanfaatan misalnya dari pengolahan, pemanfaatan sampai kemudian pemanfaatan akhir atau misalnya keuntungan ekonomi dan lain sebagainya. Karena background saya sebenarnya awalnya saya adalah sejarah, dan sejarah itu mau tidak mau dekat dengan antropologi. Bahkan ada adagium, sejarah adalah antropologi masa lalu dan antropologi adalah sejarah sekarang. Nah, artinya dekat. Kemudian saya melanjutkan sekolah dan ambil jurusan antropologi karena memang tadi tertarik saja mempelajari di alam pikiran orang-orang. Saya melihat bahwa kadang kita di balik kesederhanaan itu ada keistimewaan. Di balik kesederhanaan ada kemewahan. Di balik kesederhanaan ada keluar biasaan. Nah, itu saya tertarik. Kemudian masyarakat-masyarakat yang tinggal dekat dengan hutan, jauh dari perkotaan segala macam, itu diasumsikan mereka memiliki relasi dengan alam yang sangat baik. Serta tumbuhan itu tiap hari berelasi dengan manusia, sehingga asumsi saya karena selalu terjadi relasi antara manusia dengan alam, maka pemahaman manusia itu terhadap alam, terhadap tumbuhan itu sangat baik. Itu kenapa pilihan lokasi lebih banyak ke masyarakat yang ada di pedalaman atau dekat hutan. Kalau ke depan, saya pikir juga etnobotani harus lebih maju dalam pengertian bagaimana kita tidak hanya memahami secara mendalam perilaku atau pikiran masyarakat, tetapi juga memahami tumbuhan secara lebih holistik, lebih utuh. Misalnya, sekarang kan problem-problem kemanusiaan luar biasa ya. Ada kesehatan, ada kerusakan lingkungan, tapi ya tentunya pintu masuknya adalah informasi dari masyarakat. Kemudian setelah itu kita harus mulai bekerja sama atau membangun jaringan dengan orang-orang kimia bahan alam atau apa untuk mengetahui kandungan-kandungan tumbuhan yang tumbuhan tersebut berdasarkan informasi masyarakat diketahui sebagai obat dari suatu penyakit tertentu.
Alam semesta adalah sebuah mahakarya tak ternilai yang lahir dari tangan Tuhan Sang Pencipta. Alam menyajikan keindahan kepada manusia agar dapat dinikmati, dijaga, dan dicintai. Salah satu upaya untuk menjaga alam adalah dengan melestarikan tumbuhan yang hampir punah agar keseimbangan ekosistem di muka bumi ini tetap terjaga. Indonesia dikaruniai dengan ribuan jenis tanaman atau 1/10 dari jumlah flora di dunia. Oleh karena itulah, kita memiliki peran yang sangat penting bagi keberlangsungan hidup di bumi ini. Salah satu cara untuk menjaga keanekaragaman hayati melalui pembangunan kebun raya sebagai kawasan konservasi tumbuhan di Indonesia. Upaya tersebut sudah dilakukan sejak abad ke-19 yang dimulai oleh C.G.C. Rainward. Ia merupakan seorang botanis berkebangsaan Jerman yang menjabat sebagai Direktur Urusan Pertanian, Kesenian, dan Ilmu Pengetahuan Jawa dan pulau-pulau sekitarnya. Ia kemudian menggagas pembangunan kebun raya dengan meminta sebidang tanah di belakang kediaman Gubernur Jenderal Belanda yang kini dikenal dengan Istana Bogor. Peletakan batu pertama pembangunan kebun raya Bogor adalah pada tanggal 18 Mei 1817. Kebun raya Bogor yang kini telah berusia lebih dari 2 abad telah mengoleksi lebih dari 12.000 spesimen tanaman dan lebih dari 5.000 spesimen anggrek. Terletak di jantung kota, Kebun Raya Bogor memiliki luas sebesar 87 hektar. Lokasinya yang strategis membuat Kebun Raya Bogor selalu diminati pengunjung untuk menambah wawasan ilmu pengetahuan atau sekedar berwisata. Memasuki Kebun Raya Bogor, berdiri tegak sebuah monumen peringatan yang dibuat oleh Gubernur Jenderal Inggris Thomas Stamford Raffles untuk mengenang istrinya, Lady Olivia Mariam, yang meninggal di Bogor. Tak jauh dari monumen Lady Raffles, terdapat kolam gunting nan indah yang juga menjadi habitat alami populasi ikan air tawar, biawak, burung kowak malam, dan beberapa hewan lainnya. Masuk lebih jauh ke dalam Kebun Raya Bogor, kita akan menemukan berbagai macam koleksi taman yang indah. Antara lain Taman Meksiko. Sesuai dengan namanya, taman yang bernuansa Meksiko memiliki jenis-jenis tumbuhan iklim kering yang berasal dari Meksiko, Amerika Tengah, dan Amerika Utara. Uniknya, tanaman tersebut dapat hidup di kota hujan seperti Bogor. Ada pula Taman Theismann yang dibangun pada tahun 1884 sebagai bentuk penghargaan kepada Theismann atas jasa-jasanya terhadap perkembangan Kebun Raya Bogor. Begitu pula dengan koleksi taman lainnya yang tak kalah unik serta memiliki nilai sejarah dan manfaatnya masing-masing. Selain sebagai tempat wisata, Kebun Raya Bogor memiliki tugas fungsi di bidang konservasi dan penelitian tumbuhan dataran rendah basah. Kegiatan konservasi dilakukan untuk mencegah kerusakan dan kemusnahan dengan cara pengawetan, pelestarian, dan pemanfaatan berkelanjutan. Terdapat berbagai jenis tanaman liar yang diadopsi ke dalam lingkungan sehari-hari manusia, yakni jenis tanaman buah, umbi-umbian, dan tanaman hias. Usaha penemuan tumbuhan selalu dilakukan untuk mendatangkan manfaat bagi pembangunan negara dan masyarakat yang memiliki nilai ekonomis. Untuk mendukung fungsi penelitian, Kebun Raya Bogor memiliki fasilitas penelitian berupa laboratorium yang berfungsi sebagai tempat riset ilmiah, eksperimen, dan pelatihan. Laboratorium Truth merupakan laboratorium yang paling tua di Indonesia. Didirikan pada tahun 1844 oleh Melchior Truth sebagai laboratorium untuk ilmuwan tamu yang datang dan melakukan penelitian di Indonesia. Dari sinilah cikal bakal lembaga-lembaga penelitian di Indonesia berkembang. Lalu terdapat pula Bank Biji yang merupakan tempat koleksi biji yang berfungsi sebagai tabungan konservasi tumbuhan yang mampu bertahan hingga ratusan tahun lamanya. Sebagai kebun raya tertua di Asia, Kebun Raya Bogor tak lepas dari kawasan wisata yang menarik dan sayang jika dilewatkan. Pengunjung akan dimanjakan dengan pemandangan kekayaan anggrek Indonesia, mulai dari anggrek alam maupun anggrek hybrid, tanaman-tanaman langka seperti bunga bangkai, Rafflesia padma, dan masih banyak lagi.
Every vision begins with a spark of inspiration, and every inspiration is driven by the passion to
Tak hanya
realize
sampai di
dreams.
situ, Museum Zoologi yang ada di kawasan Kebun Raya Bogor merupakan museum fauna yang di dalamnya terdapat berbagai jenis hewan dan kerangka yang diawetkan. Salah satunya adalah mamalia terbesar di dunia, yakni paus biru. Sebagai lembaga konservasi, Kebun Raya Bogor membuka layanan identifikasi tanaman, mahasiswa magang, permintaan material, dan kerjasama bagi siswa, mahasiswa, peneliti, maupun masyarakat umum yang memerlukan data dan informasi terkait tanaman. Kebun Raya Bogor juga mempunyai program wisata flora sebagai pendukung kurikulum pendidikan dalam memperkenalkan tumbuhan dan isu-isu lingkungan hidup. Tak lupa, kami juga telah meluncurkan aplikasi Jawara atau Jelajah, Belajar, dan Wisata di Kebun Raya, agar para pengunjung dapat melakukan self-guiding selama berwisata. Alam semesta adalah sebuah mahakarya tak ternilai yang lahir dari tangan Tuhan Sang Pencipta. Alam menyajikan keindahan kepada manusia agar dapat dinikmati, dijaga, dan dicintai. Salah satu upaya untuk menjaga alam adalah dengan melestarikan tumbuhan yang hampir punah agar keseimbangan ekosistem di muka bumi ini tetap terjaga. Indonesia dikaruniai dengan ribuan jenis tanaman atau 1/10 dari jumlah flora di dunia.
Secara prinsip etnobotani itu dibangun dari dua disiplin keilmuan, yaitu ilmu antropologi dan ilmu botani. Jadi gabungan dari dua disiplin ilmu ini melahirkan atau satu disiplin ilmu, yaitu etnobotani. Kaitannya sangat erat, yaitu bagaimana memahami relasi orang dengan alam sekitarnya. Jadi relasi orang dengan tumbuhan itu seperti apa? Misalnya, bagaimana orang melihat dan memanfaatkan tumbuhan untuk obat-obatan, pangan, energi, kosmetik, kesehatan, dan macam-macam. Artinya itu hubungan orang dengan tumbuhan. Dalam hal ini biasanya pemanfaatan. Nama saya Muhammad Fathir Royani, saya peneliti etnobotani, bidang botani, pusat
Every
penelitian
vision
biologi,
begins
Lembaga
with a
Ilmu
spark of
Pengetahuan
inspiration,
Indonesia.
and every inspiration is
Etnobotani
driven by the
oleh
passion to
beberapa
realize
kalangan itu
dreams.
dianggap sebagai satu disiplin ilmu sendiri. Artinya disiplin ilmu itu dia memiliki metode. Tren sekarang sebenarnya etnobotani itu tidak hanya ke arah penggalian informasi semua jenis tumbuhan yang dimanfaatkan oleh masyarakat. Sekarang orang bisa melihat melalui satu spesies tumbuhan tertentu, kemudian didalami betul dari A sampai Z-nya, dari pemanfaatan misalnya dari pengolahan, pemanfaatan sampai kemudian pemanfaatan akhir atau misalnya keuntungan ekonomi dan lain sebagainya. Karena background saya sebenarnya awalnya saya adalah sejarah, dan sejarah itu mau tidak mau dekat dengan antropologi. Bahkan ada adagium, sejarah adalah antropologi masa lalu dan antropologi adalah sejarah sekarang. Nah, artinya dekat. Kemudian saya melanjutkan sekolah dan ambil jurusan antropologi karena memang tadi tertarik saja mempelajari di alam pikiran orang.
Every vision begins with a spark of inspiration, and every inspiration is driven by the passion to realize dreams.
Secara prinsip etnobotani itu dibangun dari dua disiplin keilmuan, yaitu ilmu antropologi dan ilmu botani. Jadi gabungan dari dua disiplin ilmu ini melahirkan atau satu disiplin ilmu yaitu etnobotani. Kaitannya sangat erat, yaitu bagaimana memahami relasi orang dengan alam sekitarnya. Jadi relasi orang dengan tumbuhan itu seperti apa? Misalnya bagaimana orang melihat dan memanfaatkan tumbuhan untuk obat-obatan, pangan, energi, kosmetik, kesehatan, dan macam-macam. Artinya itu hubungan orang dengan tumbuhan, dalam hal ini biasanya pemanfaatan. Nama saya Muhammad Fatsir Royani, saya peneliti etnobotani, bidang botani, Pusat Penelitian Biologi, Lembaga Ilmu Pengetahuan Indonesia. Etnobotani oleh beberapa kalangan itu dianggap sebagai satu disiplin ilmu sendiri. Artinya disiplin ilmu itu dia memiliki metode. Tren sekarang sebenarnya etnobotani itu tidak hanya ke arah penggalian informasi semua jenis tumbuhan yang dimanfaatkan oleh masyarakat. Sekarang orang bisa melihat melalui satu spesies tumbuhan tertentu, kemudian didalami betul dari A sampai Z-nya, dari pemanfaatan misalnya dari pengolahan, pemanfaatan sampai kemudian pemanfaatan akhir atau misalnya keuntungan ekonomi dan lain sebagainya.
Every vision begins with a spark of inspiration. Secara prinsip etnobotani itu dibangun dari dua disiplin keilmuan, yaitu ilmu antropologi dan ilmu botani. Jadi gabungan dari dua disiplin ilmu ini melahirkan prinsip etnobotani itu dibangun dari dua disiplin keilmuan, yaitu ilmu antropologi dan ilmu botani. Jadi gabungan dari dua disiplin ilmu ini melahirkan atau satu disiplin ilmu, yaitu etnobotani. Kaitannya sangat erat, yaitu bagaimana memahami relasi orang dengan alam sekitarnya. Jadi relasi orang dengan tumbuhan itu seperti apa? Misalnya bagaimana orang melihat dan memanfaatkan tumbuhan untuk obat-obatan, pangan, energi, kosmetik, kesehatan, dan macam-macam. Artinya itu hubungan orang dengan tumbuhan, dalam hal ini biasanya pemanfaatan. Nama saya Muhammad Fatsir Royani, saya peneliti etnobotani, bidang botani, Pusat Penelitian Biologi, Lembaga Ilmu Pengetahuan Indonesia.
Etnobotani oleh beberapa kalangan itu dianggap sebagai satu disiplin ilmu sendiri. Artinya disiplin
Welcome participants and speakers in room 1. We will start the session in 15 minutes.
Itu tidak hanya ke arah penggalian informasi semua
for those who are already inside, you can
oleh masyarakat.
Okay, no sound.
Sekarang orang bisa melihat melalui satu spesies tumbuhan tertentu, kemudian di dalami betul dari A sampai Z, dari pemanfaatan misalnya dari pengolahan, pemanfaatan sampai kemudian pemanfaatan akhir atau misalnya keuntungan.
Becoming a world-class institution in research, sciences, and innovation to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, LIPI. LIPI has various research centers throughout Indonesia under the coordination of the chairman Sciences, Deputy for Engineering Sciences, Deputy for Social and Humanities Sciences, and Deputy for Scientific Services. LIPI has a pivotal role in drafting and establishing government regulations and policies related to economic, political, social, and cultural aspects. LIPI also manages scientific data and scientific considerations on biodiversity conservation, as well as the publication of global indexed scientific journals, and also appointed as a scientific authority in coaching national researchers and hosting the inauguration of Indonesian research professors. To support world-class research activities, LIPI managed 5 botanic gardens as a center for ex situ plant conservation, 3 museums that have the biggest collection of flora and fauna specimens in Southeast Asia, and regional level and Indonesian natural history. Barunajaya 8 research vessel, Indonesia Culture.
Saja mempelajari di alam pikiran orang-orang, saya melihat bahwa kadang kita di balik kesederhanaan itu ada keistimewaan, di balik kesederhanaan ada kemewahan.
And Regional Training and Research Center on Marine Biodiversity and Ecosystem Health, RTRC Marbest Center.
Dengan hutan, jauh dari perkotaan segala macam, itu diasumsikan mereka memiliki relasi dengan alam yang sangat baik, serta tumbuhan itu tiap hari berelasi dengan manusia, sehingga asumsi saya karena selalu terjadi relasi antara manusia dengan alam, maka pemahaman manusia
advanced
itu
material,
alam, terhadap tumbuhan itu sangat
energy,
baik. Itu kenapa pilihan
technology, information and communication,
banyak ke masyarakat yang ada di pedalaman atau
social dynamics, humanity and culture, technology innovation and utilization of science and technology, as well as scientific services, testing and development. LIPI's national scientific collaborations include collaborative research, technology licensing, development, application, and commercialization of research results, non-research collaborations, LIPI's expertise consulting, scientific coaching for youth, students, and teachers with various universities, research institutions, local governments, and industry. As for international collaborations, LIPI conducts joint research, joint laboratories, symposium, workshops, international conferences, exchanges for researchers and experts, as well as international scholarships, while being active as a member and a focal point in various international organizations. LIPI will keep continuing to strengthen national platforms by developing world-class researchers and building scientific collaborations to make great impacts on the nation's competitiveness.
Kalau ke depan, saya pikir juga etnobotani harus lebih maju dalam pengertian bagaimana kita tidak hanya memahami secara mendalam perilaku atau pikiran masyarakat, tetapi juga memahami tumbuhan secara lebih holistik, lebih utuh. Misalnya sekarang kan problem-problem kemanusiaan luar biasa ya. Ada kesehatan, ada kerusakan lingkungan, tapi ya tentunya pintu masuknya adalah informasi dari masyarakat. Kemudian setelah itu kita harus mulai bekerja sama atau membangun jaringan dengan orang-orang kimia bahan alam atau apa untuk mengetahui kandungan-kandungan tumbuhan yang tumbuhan tersebut berdasarkan informasi masyarakat diketahui sebagai obat dari suatu penyakit tertentu.
Assalamualaikum warahmatullahi wabarakatuh. Welcome, ladies and gentlemen, participants and speakers for parallel session 1, Isibio, in room 1. Very good afternoon to everybody. My name is Ayu Safri Nurinsia, and today in this session, I will become a moderator, and I will try my best to keep the program on time. Bismillah. So for this afternoon session, we will have 12 very interesting talks, and you can see in the guideline that we already distributed to all of you. And I would like also to remind you about the conference rule. So every participant will be given 10 minutes for their talks via video presentation that we already received from the presenters. And then every 6 presenters, we will have a question and answer sessions. So for participants who already have questions, you can deliver the question via chat room in the Zoom portal. And then after the presenter finish their talks, they can directly answer the question in the chat room. Also, during after six presenters, one of the questions will be delivered by me in online. I mean live, not online. I mean live, and then the presenters will answer it directly. So for the first 6 presenters, there will be Ms. Anika Prastyawati, and then Mr. Asep Hidayat, Professor Adi Mardiastuti, Ms. Deborah Christine Purbani, Dr. Juni Setio Rahayu, and Ms. Inggit Pujastuti. Okay, because right now we already at 1 p.m. Without further ado, we will start the first video by Ms. Anika Prasetyawati. She will deliver, or in video presentation, she will deliver a talk about analysis of lactic acid bacteria biodiversity from wild honey and wild horse milk Sumbawa using RAPD-PCR and phylogenetic. So maybe the committee can help me to start the video. The floor is yours.
Good evening, ladies and gentlemen. Let me briefly introduce myself. My name is Anika Prastyawati, and I am delighted to be here today to talk to you about biodiversity of lactic acid bacteria of wild honeybee and Sumbawa horse milk by using RAPD-PCR and dendrogram. The previous title has been revised by reviewers, so I decided to perform this presentation by using a title with little change. My presentation is divided into 4 parts. I will start with introduction, and then I will look at method. Next is result and discussion, and finally conclusion. Let me begin my explaining why we did this study. Maybe some of you know about honeybee and horsemilk Sumbawa. They have many health advantages, especially in digestive tract. Honeybee, as samples of this study, has little sour taste that indicate LAB existence. As we know that LAB is grace or generally recognized as safe bacteria. However, there is still no study which explore biodiversity of lactic acid bacteria in honeybee. That's why we are interested to gain information about it in this study. We used horsemilk Sumbawa as comparison for it has been observed in previous study. To obtain information about biodiversity of LAB, there are phenotype methods have been used, such as cell morphology, carbohydrate substrate differentiation, and growth average in various temperatures. However, lack of reproducibility of those methods is the main issue. Molecular development leads us to be able to identify DNA polymorphism in genome fast, sensitively, and efficiently by using an RAPD-PCR. The basic of this method is by using some primers to multiply genome on unidentified locus, which are complement to primers used. Then, gel electrophoresis was used to detect and confirm amplification band. Here, we use also dendrogram to study similarities between isolated LIB. I will explain further in the discussion part. Next is the method. Firstly, samples were collected from 3 villages in Sumbawa, West Nusa Tenggara, Indonesia. They are Kelungkung, Sempe, and Pelak village. Then were sent to Applied Genetic Engineering and Protein Design Laboratory of Indonesian Institute of Science on ice in one-day delivery. Secondly is LAB isolation. Samples was cultured into M70 and the MRS liquid medium in reaction tube and was incubated in 24 hours. Then the aliquot of ground cultures was spread into solid medium of M17 and MRS for amount 100 microliter, then was incubated for 24 hours. Thirdly is genomic DNA isolation. Colony grown from previous steps was subcultured into medium, incubated overnight, and harvested. The pellet cells were lysed by adding lysis buffer, then purified and precipitated using buffer. Fourth step is RAPD-PCR with mixing solution containing GTG4 primers, tag polymerase, and DNA genome. Then the mixture was run into PCR machine and visualized using 2% gel agarose. Fifth step is similarity analysis using dendrogram. Electrophoresis gel photo resulted from RAPD-PCR was obtained to do a table scoring by coding acute DNA band with 1 and non-acute DNA band with 0. The scoring result was analysis using numerical taxonomy and multivariate analysis system application with Excel data to have dendrogram. Now we will look at the result and discussion. Here is the colony we obtained. From cultured samples, only samples from Kelungkung village could grow at M70 and MRS solid medium, which can be isolated as total of 40 samples and encoded SKL abbreviated of Kelungkung. For positive control, the study used LAB isolate from wild cosmic Sumbawa with total 10 samples and replaces free water as its negative control. The isolated DNA genome, which has been successfully extracted, then be identified by seeing amplification of primer GTG4 using RAPD to recognize genotype diversity, which express different phenotype. In this study, the total number of 14 isolates from Sumbawa wild honey was successfully obtained, and also from horse milk. RAPD-PCR analysis highly indicated then profile visualization of DNA. This figure performed various band size from LAB of Sumbawa wild honey and horse milk. Visualization of RAPD-PCR by applying GTG4 primer showed different genetic profiles which pointed the presence of DNA polymorphism from 25 isolates which code 8 to 21 were from Sumbawa wild honey. Samples with code 8, 11, 13, 15, 16, 20, and 21 perform similar profile compared with code number 9, 10, 12, 14, 17, 18, 19, which show different band size indicating not closely related. Isolates from Sumbawa horsemeal previously acquired by Kusnilanawati et al. Was used as positive control, and then nucleus water as negative control. Interestingly, the source of both positive control and samples were very different. The horse milk originated from dairy product, while the wild honey bee samples originated from non-timber forest product, but both of the products contained LAB. Okay, amplification profile of RAPD-PCR was further analyzed to obtain dendrogram. LIB diversity of dendrogram was displayed here. There were 5 clusters indicated different species. Cluster 1 and 2 consisted of 7 LIB isolates. 3 of them were from Kelungkung village in code 10, 14, and 17. Cluster 3 contained 11 LIB isolates originated from Kelungkung village in code 8, 9, 11, 12, 13, 15, 16, 18, and 19 of M17 medium, and also 20 and 21. Cluster 4 and 5 consisted of 6 isolates, were LIB isolated from Sumbawa horsemeal as positive control. There was group with only included isolates of wild honey and also group with positive control of horse milk only. However, there were cluster with LIB isolates from wild honey and horse milk, which showed close genetic relationship. Okay, in conclusion, let me sum up my main points. That lactic acid bacteria biodiversity from Sumbawa wild honeybee and horse milk was successfully analyzed using RAPD-PCR and dendrogram, and they result in the first amplification profile for RAPD-PCR and a 5 cluster with quite widely range of genetic diversity of dendrogram. Okay, and that brings me in the end of my presentation. Thanks for your attention.
Yes, thank you very much, Ms. Anika Prastyawati. For our second presenter, we would like to welcome Mr. Asep Hidayat. He will present about diversity of endophytic fungi isolated from benzoin-producing tree, Styrax sumatrana. I hope I spell it correctly. So the floor is yours, Mr. Asep Hidayat, via video.
Thank you, moderator, and good afternoon, everybody. In this time, I would like to explain of our research. The title research is diversity of the endophyte fungus isolated from the benzoin producing tree, Styrax sumatrana. My name is Asep Hidayat. I am from Forest Research and Development Center, Ministry of the Environmental and Forestry. Now, let's me to introduce the background of this research. The Styrax sumatrana is one of 130 species belonging to genus Styrax in the Styraceae. Styrax sumatrana is the endemic tree from the Indonesia and widely distributed in the North Sumatra province. This tree can produce kemenyan or benzoin resin. This resin will secrete when the bark injured. The resin also can use as the incense and make them high economic value, and not timber forest product resources. This resin as the raw material for many purposes, such as cosmetic, perfume, drug, and other. Kemenyan resin from the Indonesia so different to other because this resin is very unique, especially from the free cinnamic acid and a cinnamic derivative product. Kemenyan or benzoin resin containing 130 compounds and potential as bioactive. The main compound of benzoin resin such as phenolic, terpenoid, and lignan and saponin. Fungal endophyte are known to produce various bioactive activity, including benzoin production syrup benzoin, and might contribute to the host resin production or properties. Unfortunately, fungal endophyte associated with the Syrax sumatrana were less in the study. According to the background of this research, the objective of this research was to evaluate distribution and diversity of the culturable fungal endophyte associated with the three part, bark, stem, and leaf of the Syrax sumatrana. Let's move to material and method of our research. In this slide, also the general of our method. Our method containing the sample collection, isolation and purification, fungal identification, and diversity analysis. Sample was collected from 3 locations in North Sumatra. Isolation and purification was conducted in the agar medium such as potato dextrose agar, yeast dextrose agar, yeast malt extract agar. Fungal identification was conducted using DNA analysis. Diversity analysis was conducted and focusing in the important parameter such as Simpson-Wiener index, Simpson diversity index, uniformity index, Simpson dominant index, Margalef index, and special richness index. Now I would like to explain the result of our study. From the collection sample, we collected 11 trees of the Syrax sumatrana collected from 3 locations of the local forest community in the Simalungun, Dolok Humbang Hasudutan, and North Samanuli. 464 different part organ of the Syrax sumatrana was cultured to the agar medium, and we obtained 31 purified isolate. From this isolate, 15 isolate come from bark, 8 isolate come from the stem, 8 isolate come from the leaf. In this slide, show the morphology characteristic from our pure isolate. Coloniform, mycelial color, reverse media color, so different among all of isolate. Based on the DNA identification, all of the isolate of the endophytic fungus was identified into 2 classes, 6 families, 10 genera, and 70 species. Majority of isolate was isolate from the bark. We obtained 15 isolate. All of fungal species classified as the member Ascomycota. In this slide, show the relative abundance of our isolate. The high total abundance was for Fusarium genus, followed by Pilostica, Pestatiopsis, Neopestatilosis, and Trichoderma. Fusarium genus was the most abundant genus in the bark and leaf, while in the stem, Pilostica as the most abundant. Genus Fusarium is known as the parasite and also reported to have resistance against pathogen as well as anti-cancer property and anti-inflammatory, anti-tussy, anti-allergy properties. In this slide also show the diversity index of endophytic fungus isolated from the Syrax sumatrana. As you can see in this slide, that endophytic fungus species show variation distribution in Syrax sumatrana plant organ. But has the richest fungus species and most abundant in the term of the fungal isolate. Diversity index, Samson-Wiener, Simpson, and dominant index was determined. The value of the Samson-Wiener index of endophytic fungus was high in the bark, followed in the stem and in the leaf. The similar result also was observed in the for Simpson index, where the bark had the high value, followed in the stem and leaf. In general, the height of the Samson-Wiener index value was closely related with Simpson index value to become 1. Dominant index saw the low value in the bark compared to the stem and leaf. Dominant of the endophytic in the leaf is greater than in the bark and the stem, with just 2 genera were found, Fusarium and Neopeptatheosis. The Simpson index, fungal in the leaf classified into small, while in the bark classified in the moderate, and then in the stem classified in the high uniformity. Endophytic fungus community from different isolate part indicated the structure, richness, diversity, and dominance of the different species, as well affected by plant organ. Previous studies suggested that morphology and chemical substrate in the host tissue also affected by endophytic fungal community composition in the plant root and stem. According to the result of this research, the conclusion of this research are a total of 31 cultural by endophytic fungus isolates were a success obtained from different parts of the Syrax sumatrana. High diversity and significant endophytic fungus from the plant organ specificity were observed in Syrax sumatrana. Bark was most diverse and had the high isolation frequency among other organ. The current study provides initial information on the endophytic fungus diversity, community composition, and in-organ specificity in the suborgan of Syrax sumatrana. Bioactive compound produced by this fungus as well as it related to the resin production are to be future investigated. We would like to express my sincere thanks to my office, Ministry of the Environmental and Forestry, and Ministry of the Research and Technology, or National Research and Innovation Agency, for finding support of this research. That's all. Thank you very much for your kind attention.
Thank you very much, Mr. Asep Hidayat. So before I proceed to the next presenter, I would like to inform for all participants and speakers, you may deliver questions to speakers via chat room, and also presenters can directly answer the question given in the chat room. So now we proceed to the next presenter. I would like to welcome Professor Ani Mardiastuti, and she will deliver a presentation about traditional uses of herpetofauna among local people in Sumatra, Indonesia, conservation implication. The floor is yours.
Good morning, everybody. Thank you for giving me an opportunity to present our study. My name is Ani Mardiastuti, and on behalf of my colleague in the Faculty of Forestry and Environment IPB University, I would like to present our research entitled traditional use of herpetofauna practiced by the local people in the island of Sumatra, Indonesia, implication for conservation. We might often heard about ethnobotany, but maybe less heard about ethnozoology. This is because there has been only a few ethnozoological studies that has been conducted in Indonesia. Although the local people in Indonesia were in fact used animals to fulfill their daily needs, including for consumption, traditional medicine, and as well for traditional rituals and myth. Among the wildlife animals, reptiles and amphibians, or collectively known as herpetofauna, were widely used. We focused our research study in Sumatra because this island has been undergone a rapid deforestation and habitat fragmentation, as seen in this slide. Human-wildlife encounter is also more intense because humans potentially live nearer to wildlife species. Hunting for wildlife would be much more easier in the fragmented and deforested area. So the objective of the study was to provide an overview of the traditional use of herpetofauna by the local people in Sumatra and analyze its implication for conservation. We conducted this study, collect all relevant literature from internet, and analyze all information by calculating relative frequency citation, basically how often a certain topic was mentioned in the reference, and also check the conservation status of species being used. For the conservation status, we use IUCN Red List, CITES Appendix for international trade, and protection status by Indonesian law. Literature regarding ethnozoology in Sumatra was actually rather limited, only 16. For ethnoherpetofauna was much more limited, only 11 literatures, dated back to 2009. We could not find any literature before 2009, although there could be some literatures, but not available through internet. In all available literatures, there were 22 herpetofauna species used by the local people, consisted of 18 species of reptiles and 4 amphibians. You can see here in the pie chart that for reptiles, basically all group of reptiles were being used by the local people in Sumatra, including snakes, lizards, freshwater turtles, sea turtles, and even crocodiles. We also tried to collect information on why the local people hunt and use the herpetofauna. In addition, for consumption, the herpetofauna were also popular as traditional medicine for various diseases, ranging from digestive diseases, musculoskeletal disease, respiratory diseases such as asthma, eye diseases, and many other diseases. As parts for being used, basically all parts were useful. The whole body, meat, skin, blood, bile, head, teeth, and even urine and feces. 2 most widely used species were reticulated phyton, Malaya Phyton reticulatus, and Varanus salvator, the water monitor. Other commonly used species were turtles, namely Asiatic softshell turtle, Amida cartilaginea, and Malayan softshell turtle, Dogania suplana. Crocodile, as I mentioned before, surprisingly were also popular. Among the species that have been used by the local people, 8 species were already listed as threatened species. 1 species, the Penteterapin, Batagor borneoensis, has been listed as critically endangered. This is the highest status of the IUCN list. The species was still, unfortunately, harvested by the local people. There's a possibility that the local people was not aware that these species, as well as many other, were in fact protected by Indonesian law. About 32 of the species were already considered as threatened species, either critically endangered, or vulnerable by the IUCN Red List. These species were already protected by Indonesian law, thank God. Yeah. Most of the threatened species and protected species were turtles and snakes. As the local people might not aware about the conservation status of the species that they harvested, we need to have an awareness program, especially for the species having low population number. From this research, we can conclude that there were not many research focused on ethnozoology and ethnoherpetology, although herpetofauna has been commonly used by the local people since a long time ago. There were 22 species of herpetofauna that were used by the local people, mainly for traditional medicine. Of the 22 species commonly used, 8 species were threatened, and thus we need to have an awareness program for the local people. In addition, we also need to have further studies covering other important taxa, mammals and birds and many others, and in other geographical regions within Indonesia, not only in Sumatra. That's all from us. We thank the Ministry of Research and Technology, National Research and Innovation Agency, the Government of Indonesia for funding this research under the scheme of Penelitian Dasar Unggulan Perguruan Tinggi. Thank you very much.
Thank you, Professor. Very nice presentation and very informative. And I would like to again remind you, if you have questions to the presenters, to the previous presenters, you can just type your question in the chat room, and then we welcome the presenters to answer directly the questions in the chat room also. So right now we will welcome the next presenter, Ms. Deborah Christine Purbani, and she will present via video about spatial diversity of microalgae in Simelu Island, Indonesia. The floor is yours.
Good afternoon, my name is Deborah Christine Purbani. I'm from Indonesian Culture Collection Research Center for Biology LIPI. In this opportunity, I would like to present our research, Spatial Diversity of Microalgae in Simelu Island, Indonesia. Diversity of Microalgae in Simelu Island, Indonesia. Firstly, I would like to explain about microalgae. Microalgae are a vast group of prokaryotic and eukaryotic phototrophic microorganisms which present in unicellular or simple multicellular and occur in all natural water bodies. They play a major role to be a producer of aquatic ecosystem as they contain a rich source of protein, carbohydrate, and essential fatty acid. Therefore, microalgae become important aquatic microorganisms for energy, livestock, health, food, and cosmetic sector. As a tropical maritime country, Indonesia has a high sunlight intensity. It means that the presence of microalgae in aquatic resources could be potentially more diverse. However, the information regarding the diversity of microalgae from Indonesian natural resources is very limited. Therefore, the exploration of microalgae from Indonesia resources need to be continued in spatial scale. Conducting spatial isolation is essential that may imply the microalgae community structure and biodiversity sustainability, which also benefit for the future in energy, livestock, health, food, and cosmetic sector. The present study was carried out to isolate and identify microalgae from one of outer island, Simelu, which is located in Indian Ocean, 105 nautical miles from Melabuh, Aceh, Sumatra mainland. This region has a wide natural aquatic ecosystem to view the pattern for spatial macroalgae diversity in fresh and seawater. By isolating this microorganism, we expected to obtain macroalgae culture as a national treasure of biological resources and promising use in the forthcoming. Water samples were collected from different habitats of 4 locations in similar island Indonesia, namely Langi Beach, Batu Alavan Beach, Kuala Makmur River, and Laut Tawar Lake. For the macroalgae collection procedure, we applied the random sampling method and measure for a physicochemical parameter, including water temperature and pH from the surface and bottom of the water of its sampling site. Macroalgae were then isolated using the capillary micropipetting method under the Olympus-like microscope, and then purified and cultivated under like condition in AF6 freshwater and IMK marine liquid medium. The macroalgae cultures selection was based on their growth, morphology, and homogeneity. We perform a microscopic observation under light microscope to select it and identify the macroalgae culture. The taxa identification was referred to standard taxonomic manual book by observing the morphological form of chloroplast, filament, the shape and size of cell. Molecular identification was performed by preparing macroalgae from freshly grown cell of the liquid medium. DNA template was extracted by using the genomic DNA minikit plant geneate following the manufacture protocol. Macroalgae cell, the lysis macroalgae cell was used for PCR amplification of the partial 18S rRNA gene with primer 18SF and 18SR using the Gotak master mic. The PCR amplicon were then visual on 2% agarose for confirmation and sequenced to first base Malaysia. The trace of amplicon from forward and reverse primer were aligned to ChromaSpro to determine the 18S rRNA gene sequence. Subsequently, basic local element search tool for nucleotide depth was carried out to compare the homology among other type species on public database. Meanwhile, to figure the phylogenetic tree, all sequences were aligned using Crystal W, and the distance metric was calculated using the 2 parameters of Kimura method on Mega 10 software. Finally, the tree was constructed by applying the network joining with 1000 bootstrap welding method with Chilomonas paramecium as an outer group. A total of 17 strains of macroalgae were isolated and identified from water sample in similar island Indonesia and grouped into green algae based on the morphology observation. This was the first step for grouping macroalgae, which the taxonomy placement was listed on table 1. Molecular approach supported for more certain in lower level taxon, and they were taxonomically belong to Chlorella casrelli, Chlorella sorokiniana, Oocytis heteromucosa, Stichococcus basilaris, Cocomixa melkoniae, Scenedesmus acuminatus, Scenedesmus bijucus, Coelastrella oocytis formis, Anchisrodesmus falcatus, Chlorococcum oleifacien, Stichioclonium tenue, and Planopilla laetiferens. They had a high homology consensus in range of 99 to 100% compared to the type strain in the public database. The phylogenetic tree emphasizes to taxonomy placement of the obtained macroalgae culture, which were related to 12 species in 8 clades. And each of isolate macroalgae has a unique character and function. Members of Chlorophyceae were predominantly found among all culture microalgae, which no single species possesses a higher density than other, meaning that they presented in equal density through the water samples. This result reflect that similar aquatic ecosystem were in the healthy condition, providing resources to many microalgae. Meanwhile, almost all of those species were found in different habitat and location, meaning that each species found in freshwater or seawaters. The growth of microalgae on aquatic ecosystem is supported by the physicochemical properties such as temperature and pH. The water temperature on site location was its range of 25 to 30 degrees Celsius and pH from 6.9 to 8.1. This condition can support the life processes of the aquatic organism living with the optical temperature for microalgae in general between 24 and 28 degrees Celsius, although this may vary to the type of the species and strain. On the other hand, the pH range for most microalgae is between 7 and 9, with the optimum in 6.5 to 9.5. The pH was slightly higher in Kuala Makmur rivers, which is maybe attributed to the increased photosynthetic activity in the aquatic bodies and demanded more CO2 than furnished by respiration and the decomposition. Therefore, pH is the important quality parameter that could affect the survival and nutrition of biological life in water community. This exploration, isolation, and preservation of microalgae are essential to be conducted to unravel the biological resources of Indonesian archipelago, which has many potential benefit of various life aspects. Microalgae could provide another perspective for answering the future challenge in science, innovation, and technology development. Furthermore, delineating the microalgae biodiversity could impact on reserving natural resources in Indonesia, which become a big challenge for sustainable development. In conclusion, microalgae isolated from single island Indonesia filled the spatial diversity in fresh and seawater from various habitat and location. They consist of three major classes, Spiropelales, Chlamydomonadales, and Chaetoporales, which were closely related to Corella casrelli, Corella sorokiniana, Oocytis heteromucosa, Stichococcus basilaris, Coccomixa melkoniae, Scenedesmus acuminatus, Scenedesmus bijugus, Coelastrella oocytis formis, Anchisrodesmus falcatus, Chlorococcum aleofascien, Stygeoclonium tenue, and Planophylla laetivirens. This information would be an important biodiversity hotspot as genetic resources for taxonomic study and bioprospect in the future. Thank you very much for your great attention. May all those we have shared be beneficial for all of us.
Thank you very much, Ms. Deborah. Very interesting presentation. And I can see from the chat room there are questions already written here. Question for Ms. Anika, the first presenter, and then question for Mr. Asep, the second presenter, also for Professor Ani. And I can see also here that Professor Ani already respond to the questions. So for all participants that have questions to the presenter, you can type your question in the chat room. And presenters, please, you can also answer the question here in the chat room. So now we can proceed to the next presenter, Dr. Yuni Stio Rahayu. She will present from a video about nutrient input from the litter fall in the lowland forest of Mount Gede Pangrango National Park. The floor is yours.
Ladies and gentlemen, good afternoon. It is a good opportunity for me to deliver a research presentation with the title Nutrient Input from the Litter Fall in the Lowland Forest of Gunung Gede National Park. My name is Yunis Jorahayu from Research Center for Biology. As well known that land conversion is one of the reasons for the declining of forest area and soil quality, and the land conversion mostly due to the conversion of forests into agricultural land, plantations, illegal logging, hunting, mining, forest fire, and the other purposes. Land conversion will affect to the nutrient cycles in the ecosystem, soil fertility, and the biodiversity. Nutrient cycle has an important role for the plant's growth. The nutrient which return through the litter to the forest floor is the main pathway for the transferring nutrient from plants to the soil. Through the process of decomposition of organic matter, litter will release nutrient into the soil and subsequently taken up by plants for producing new biomass. At the ecosystem level, nutrient supply is important in increasing fertility and the productivity of tropical forest ecosystem, which is generally known to have low soil fertility. Forest area in Indonesia is estimated at about 94.1 million hectares, and the most declining forest ecosystem area was lowland forest ecosystem in the period of 1980 until 1970. The remaining lowland forests in Java are mostly in conservation area. Mount Gunung Gede National Park is one of the remaining lowland forests in West Java. Decrease of the forest area in some forest ecosystem affected to the reduce of the nutrient input to the ecosystem as the main nutrient source to the environment. Therefore, the purposes of the study was to determine the nutrient return in the lowland forest of Mount Gede National Park. Our study site is in the lowland forest of Mount Gede National Park, West Java, with the altitude was 822 meters above sea level. 1 hectare permanent plots were established to determine the tree structure and composition, and the dominant species in the study site were Mysopsis eminii, Sima wallicii, Dixosilum, Neoclea, and Ternandra, with the basal area was 23.2 meters square per hectare. 20-liter traps were randomly installed within permanent 1 hectare plots, and each was placed in 1 meter above the ground and tied up to 4 1-meter long PVC poles stuck into the soil. And the litter component was separated into leaf litter, twig and woody litter fall, stem more than 2 centimeter diameter, reproductive part, other material that could not be classified, and this litter fall were collected monthly. Digested litter samples were used to measure phosphorus, calcium, magnesium, potassium, natrium content. Now we are going to the result in discussion about the litter fall nutrient content. The result shows in this figure that most of component of litter fall release nutrient, potassium, calcium, magnesium, and sodium content was higher in the rainy season than that of dry season. This predicted due to the lack of water in the dry season reduced the absorption of nutrient from the soil. The high nutrient return during rainy season was in line with several studies in tropical forest regions and the broadleaf wet mountain forests. This is also supported by research of researchers who are described that nutrient leaching from the litter during the dry season will increase the availability of plants nutrient at the beginning of the rainy season. And therefore, the result shows that the rainy season higher than dry season. These results also correspond to the total number of litter fall during rainy season, which is higher than dry season. It was estimated that all plant species contribute to the hike of litter fall in rainy season. And the nutrient return to the forest floor was varied among components. It's mostly due to the litter quality of the litter component and the composition of dominant species. This phenomenon similar to the results have been done in some tropical forests. If we talk about the potential return through the litter fall, the total annual return from the highest to the lowest were as follow: calcium higher than nitrogen, higher than potassium, and this is also higher than magnesium, natrium, and phosphorus. These nutrient order are the same as recorded in tropical rainforests in China and secondary lowland forests. However, this sequence is different when compared to the peat swamp forest in central Kalimantan. The difference in annual nutrient return in some forest ecosystems are mainly due to the rainfall, temperature, water, nutrient availability in the soil, composition of plant species, and the litter production. Related to the order of nutrient return of each litter fall component is in accordance with the order of the production of litter fall component, where the leaf litter fall component has the highest value compared to the other components. And the leaf component is the biggest contributor to the annual nutrient return. This is positively correlated to the litter fall production, where leaf litter production in the lowland forest of Gunung Gede Pangrango National Park was recorded for 78%. And some research also reveal that leaf litter is the largest contributor in the return of nutrient to the soil in various types of ecosystem. And phosphorus content in lowland forest is lower than the other macronutrients. Low phosphorus nutrient returns indicate that the phosphorus is a limited factor for plant growth, both in the tropic and subtropical region. This subsequently affects to the low phosphorus soil availability. Going to the conclusions, the research on nutrient dynamic in the lowland forest of Gunung Gede Pangrango National Park has shown high nutrient return occur during the rainy season, except for phosphorus. And in regard to the leaf litter component, leaves are the main source of nutrient return to the soil. Successive nutrient return from the highest to the lowest order is calcium, higher than nitrogen, higher than potassium, magnesium, phosphorus, and the sodium. These nutrient returns are lower than other forest ecosystems in tropical rainforests and subtropical plantation forests. The lowest nutrient return of phosphorus indicate that phosphorus is a limiting factor for plant growth. We would like to thank the head of Research Center for Biology for supporting the research and the head of Gunung Gede National Park for providing research opportunity in the national park. And thank you for our college who have supported through the research. Thank you.
Thank you very much, Dr. Juni. Very interesting presentation as well. So we will have our last presenter for the first six speakers. So after this sixth speaker, we will have a question and answer session, but probably only one question will be delivered live, because we have very tight schedule today. So for the next presenter, we will welcome Ms. Inggit Pujiastuti, and she will present from video also about flowering and fruiting of Sarcotheca macrophylla, BL, an endemic species of Borneo, and implication for conservation. The time is yours.
Flowering and fruiting of Sarcotheca macrophylla, an endemic species of Borneo, and implication for conservation. Introduction. Flowering and fruiting are critical life story states for plant conservation. Sarcotheca macrophylla is an endemic species of Borneo. Little is known about the flowering and fruiting of this endemic species. The present study aims to investigate the flowering and fruiting of Sarcotheca macrophylla in terms of the first-time flowering, morphology of the flower and fruits, and number of flower of fruit. Method. The time this research was conducted started from 2015 and continue until December 2019. The location in Bogor Botanic Garden Nursery, Indonesian Science Institute of Science. The objective of this research is seeds of macrophylla that were sown to generate seedling. The seeds include 25 seeds from Katingan, Central Kalimantan in 2015 year, 10 seeds from 2006 from the Bogor Botanic Gardens collection plant. After that, 10 seeds in 2017 and 15 seeds in 2018 and 20 seeds in 2019 from plant grow from seeds sourced from Katingan, Central Kalimantan. The parameter is number of seedlings and when the seedling produce flower for the first time. B. The number of seedling and when the seedling produce flower for the first time. B. Morphology of flower, fruit, and seed. C. Number of flower, fruit, and seed. Result. First time of Sarcotheca macrophylla flowering. You can see the table. Then in the first with number seed 8, the flower of seedling with initial flower 8, 100%. The age of seedling year of the seed sowing, 7 and 10 months in 2015. Second, for seedling, only 2 seedlings initial flowering, and the presentation is 50%. The age is 5 months until 1 year, 4 months in 2016. 3, 10 seedling, only 4 seedling with initial flower. The presentation in 40%. The age of seedling is 6 until 8 months in 2017. Then 4, 15 seedling, only 3 seedling with initial flowering. It means it's 20% Then the age is 7 until 1 month and 2018. And 5, 10 seedlings, only 2 seedlings with initial flowering. It means that 20%, the age of seedling 7 months and 2019. The morphology of inflorescence and flowering. Inflorescence type is panicle with several flowering clusters. Inflorescence length is 10 until 20 centimeters. Number cluster and 2 until 8 cluster with 0 until 10 flower per cluster. Location in terminal and axillaries. The flowers is dark red, sepal colored obovates, anther ovates, petal obovates, oblong to lanceolate. The figure is the one is cluster of flower of mark. Then the second flower arise from the edge of stem terminal, and flower arise from the petiole and axillaries. Then 3 is a morphology of flower of marcotyl calyx. E, stigma B, corollary and pistil D. The morphology of fruit and seed. The fruit of Sarcotheca macrophylla produce following the flower filtrate. Each individual has 2 until 8 fruit plus cluster. Each fruit cluster consists 1 until 10 fruit. Fruits red maroon when unripe and become black purple when ripe. Each fruit consists of 0 until 3 seeds. Seed, ellipsoid, black, green. Figure 4 is a fruit cluster of Sarcotheca macrophylla, and figure 5 is each fruit cluster consists 0 until 10 fruits, and B is a morphology of fruit and of Sarcotheca macrophylla. And figure 6 is morphology of Sarcotheca macrophylla seed. Discussion. Sarcotheca macrophylla precocious flowering time. Researchers et al. Reported that generally plants begin flowering at 3 until 5 years after germination. However, some species have to be able of profuse flowering by producing initial flowering at the seedling stage, such as citrus seedling at the beginning flowering at 3 months until 2 months after germination. Terminalia tabula seedling that begin flowering at 3 months after germination. Proposis seedlings that begin flowering at 3 until 4 months after germination. The present study will show that the average Sarcotheca macrophylla conduct precocious flowering at 5 until 16 months. Sarcotheca macrophylla precocious cause factor. The flowering of morphology can be influenced by one of these factors and interaction of the factor. Further research is required to investigate factors influencing the flowering of Sarcotheca macrophylla. Sarcotheca macrophylla precocious flowering implication for conservation. Information about precocious flowering is widely used for the research and breeding of the flowers. And the second, Respirating using precocious plant is also useful for accelerating natural regeneration and for consuming wildlife. Conclusion. Seedling of Sarcotheca macrophylla at Bogor Botanic Gardens were able to produce precocious flowering at the age of seedling of 5 months until 1/6 months. The precocious flowering of the seedling lasts for 1 until 2 months. Thank you.
Thank you very much, Mrs. Inggit Pujastuti. So now we will start our question and answer for the first session. But since we only have 5 minutes, I think we will have only one question will be given live from the chat. Okay, actually, I want to deliver the question for Dr. Yuni, but since she already answered one question for Prof. Ani, Miss Yuni, Dr. Judy, Theo, are you there?
Yes.
Okay.
Yes, I am.
Okay, thank you. There are 2 questions from the participants for you. One already answered by you in chat, but one probably, can I give it via live? Yeah, Bu, ya?
Yeah, okay.
Okay, so it's a question from Dr. Mukhtar Davis from RC Biology. I will just read it. So, nice and interesting presentation. I am not familiar with this field type experiment. Could you explain why use hanging trap? Why not just collecting the litter from the forest floor? Secondly, using litter trap, obviously plants below 1 meter are excluded. How can one minimize the bias of collecting litter only from trees with high stature plant and large canopy? So the floor is yours, Dr. Yuni, to answer.
Okay. Thank you very much for the question. And actually, the question is almost similar with Bu Ani questions. Why we use a litter trap? Because we want to know the nutrient input freshly from the litter fall. This is the point, very important point in the forest, in the nutrient cycles. Because if we know the nutrient input, so we know the how much nutrient will be used for the to this ecosystem. And then this method already standardized. So this is internationally used for the size 1 and 1 square meter. But the samples in 1 hectare plots, the litter traps should be installed less than 20 traps. So 20 traps, it's enough for 1 hectare. This is already international standard method. After we collected, and then we separated the samples, and we will estimate for in 1 hectare plots. This is very important things. I think, is it answer the questions?
I think Dr. Mukhtar Davis also here. Are you satisfied with the answer, Dr. Mukhtar?
Hello, can you hear me?
Yes, yes, perfectly.
My concern is because you are using a hanging trap that is 1 meter above ground, so it only will catch the litter from like high trees. My question is
okay.
Like the one that I post in written. Why? How can one minimize the bias of just only collecting the litter from high trees?
Okay.
Yeah,
I got your point. So this is a different measurement. One, this is for the litter fall from the trees, and the other that you mentioned is from the below, what we call it, understory, like understory species. We measure by using 50.50 centimeter square in 40 in 40 places in one hectare. So it's different. But in this presentation, I only use for the litter fall from the trees.
Okay, I see. Thank you.
Yeah.
Okay, thank you very much, Dr. Yuni. So I think the conclusion is, if you have a question and you want to answer the question perfectly, then you need the tools that match with those things. So you cannot use this method to answer a different question. I think sort of like that,
yeah. It's correct. But
okay, thank you very much for the all 6 presenter. And now, just on time, we will start the second 6 presenters. So we will have Ms. Fatima, Dr. Ian Robiansyah, Mr. Izhar Khairullah, Dr. Yunis Tiorahayu again, and Mr. M. Yunus, and also the last one is Mr. Kurniawan. So without further ado, I will welcome the video presentation of Ms. Fatima, and she will deliver about the potency of phosphate solubilizing bacteria in the soil of Tuban Mangrove Center, East Java. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. It is an honor to meet you here. Before I begin, let me introduce myself. My name is Fatima. I'm from Universitas Airlangga. In this opportunity, I would like to present a small part of our research entitled The Potency of Phosphate Solubilizing Bacteria in the Soil of Tuban Mangrove Center. This is our team. Okay, we are going to introduction. Phosphor, is one of the macronutrient for the metabolism of plants to form ATP. Phosphor is available between 0.01 until 0.1% among the whole compounds in soil. The availability of phosphate in soil is abundant. Unfortunately, plants cannot utilize 19.5 until 19.9 of it because of the insoluble form of the phosphate. Plants barely absorb bonded phosphor, therefore it must be turned into an absorbable form. To increase the availability of phosphor for plants, large amount of fertilizer are used on a regular basis, but after application, a large proportion of fertilizer phosphorus is quickly transferred to insoluble form. However, a chemical fertilizer may have a negative effect, such as include waterway pollution, soil acidification, and mineral depletion to reduce the harmful effect of chemical fertilizer on human health and environment. And nowadays, there is increasing trend on the utilization of biofertilizer. Biofertilizer are usually prepared as carrier-based inoculant containing effective microorganisms. One of the role of microorganism in biofertilizer is phosphate solubilizer. This microorganism capable to change inorganic phosphate into organic phosphate available for plant enzymatically by using phosphatase enzyme. There are plenty of microorganisms in soil to solubilize phosphorus, include yeast, mold, and bacteria. Therefore, we focus to obtain Indonesian local isolate of phosphate solubilizing bacteria for biofertilizer candidates. Now we go to materials and methods. The soil samples were compiled based on the pH similarity. Then 25 grams of the samples put into 225 milliliter sterile physiological saline solution. The soil suspension was homogenized with shaker for 30 minutes, left for 15 minutes, then dilute until it reaches 8 times serial dilution. And then 3 serial dilution consisting 1, 5, 8 times serial were poured respectively into Pekoff Kaya media. The plate incubated at room temperature for 3 until 5 days, and the phosphate solubilizing bacteria will exhibit the ability to solubilize phosphorus on the Pekoff Kaya medium by showing the clear zone formation around the growing bacterial colonies. The growing colonies were isolated and purified in nutrient agar medium. This is a potency assay of phosphate solubilizing bacteria. The ability of the pure isolate to solubilize phosphate was re-examined on plate filled with a Pekoff Kaya agar medium by using the spot inoculation method. The potential test for each isolate was conducted 3 times. Okay, and the clear zone diameter formation around colonies was measured, and the obtained data were calculated and stated as phosphate solubilization index or PSI in the following formula. The isolate with the highest PSI number were then characterized morphology, biochemical, and molecularly with 16 srRNA gene to obtain the species name. Table 1 shows the data of 19 isolates of phosphate solubilizing bacteria that were successfully grown on a selective Pekoff Kaya agar medium. There are 11 isolates in location 1, pH 6, and 7 isolate in location 2, pH 6, and 1 isolate in location 1, pH 4. The test result of 19 isolates on Pekoff Kaya medium is presented in figure 1. We can see yellow arrows indicate bacterial colonies, and red arrows indicate the clear zone formed. Pekoff Kaya medium contain phosphate, in the form of compound calcium phosphate, and the compound is bonded by bacteria and released by H2PO4- ion. Binding of compound and the release of ions cause the appearance of clear zone around the colony. The clear zone is formed because of the phosphatase enzyme enzyme ability. The clear zone occurs because of pH decreasing in a medium as the result of the phosphatase enzyme activity. Table 2 below present the result of calculating the phosphatase solubility index of each bacterial isolate, and isolate 4A have the highest PSI, that is 2.36. This is the macroscopic and microscopic characteristic of the phosphate solubilizing bacteria isolate from Tuban Mangrove Center. Okay, in the right side, we can see the figure macroscopic characteristic of isolate 4A. In this table, we want to discuss or say about a physiological and biochemical tests using the microbag 24A kit on the most potential isolates. In this test, only 4A isolate was tested, which is isolate with the highest solubilization index. And the result from physiological and biochemical tests are presented in table 4. As we can see in this table, comparison of 4A and Klebsiella are only different in the result of the adonitol test and sucrose fermentation. Yeah, red line. The result of macroscopic, microscopic, physiological, biochemical, and molecular identification show that 4A isolate belongs to the Klebsiella sp. With a similarity index of 93%. This is calculated by Jaccard index and identified as Klebsiella varicolastrin DBPP1 with similarity 97.28% by 16S rRNA gene sequence analysis. This is the final slide, or the last slide. Based on the result, it can be concluded that the phosphate solubilizing bacteria isolated from Mangrove Center Tuban were 19 isolate. The 4A isolate has the highest potential among the others, and 4A isolate has similarity as much as 7.28% to Klebsiella varicolastrin DBPP1. That is my presentation. Thank you very much for your great attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Ms. Fatima. And for participants who have questions for Ms. Fatima or for previous presenters, you can give questions in the chat room. And I can see here there is a question for Mrs. Inggit Astuti. So maybe you can directly answer the question via chat room. Now we will proceed to the next presenter, Dr. Ian Robiansyah. And from video presentation, he will deliver the theme of presentation about population and conservation status assessment of Ettlingera balikpapanensis, an endemic ginger in East Kalimantan. The floor is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for the Isibio committee for letting me present the result of our studies with the title of Population and Conservative Status Reassessment of Ethlingera balikpapanensis, an endemic ginger in East Kalimantan. For the introduction of our presentation, the Balikpapan ginger or Ethlingera balikpapanensis is a ginger species endemic to East Kalimantan province in Indonesia. The species is known only from two locations near Balikpapan city and is threatened by forest encroachment and forest fires. Olsons and Olander in IUCN Red List website consider the species is endangered based on the criteria of B2A, B3, based on a small area of occupancy, number of locations less than 5, and continuing decline in area, extent, and quality of habitats. However, the assessment was based only on herbarium records, and there is no information on the current population status of the species. Therefore, the objective of our study is to assess the current condition of the distribution, population size, and threats of the Ethlingera balikpapanensis, and also to reassess its extinction risk based on IUCN Red List category and criteria. This is the study sites of our study. It is located in Kebun Raya Balikpapan, and also some area in Sungai Wen Protected Forest. For the population survey, we use focus or intuitive control survey methods. When the clump of Ethlingera balikpapanensis was found, some population variables were measured, such as total number of individuals within the clump, height of each individual, and also width of the clump. For the environmental variables, the following variables were also measured: elevation, slope, aspect, and canopy cover. To see the threats to the species, we also estimate the tree cover loss of the habitats using Global Forest Watch website. After collecting the data, we also try to reassess its conservation status based on IUCN Red List category and criteria. And this is the result of our study. During the survey, we found 14 clumps with a total of 171 individuals in the study site. Most of the individuals have 3 to 3.9 height, 3 to 3.9 meter height, with the highest individual is around 6 meter height. According to Hadia et al., who conducted a survey in Teleng Subdistrict, they found also around 150 individuals of Ethlingera balikpapanensis. With this additional data, the total of individuals of Ethlingera balikpapanensis in East Kalimantan is estimated to be around 321 individuals. This is the results of the condition of each clump of Ethlingera balikpapanensis in our study site. We found, as I mentioned before, 14 clump with the total individuals within clump range from 4 to 26 individual. The widest clump is around 15 meters wide, with the elevation range from 38 to 49.8 meter above sea level. The slope of the slope of the habitats for the Ethlingera balikpapanensis was ranged from 1 to 32 degrees, with the canopy gap is around 27.4%. According to Global Forest Watch, the threat to the species from habitat conversion and degradation in East Kalimantan, it lost around 2.6 million hectares of tree cover from 2001 to 2019, equivalent to 23% decrease in tree covers since 2000s. The highest tree cover loss was observed in 2016 with around 300,000 hectare tree cover loss. For the conservation status reassessment, for the criteria A, we can assess the species is net threatened as the habitat loss is estimated to be around 30%. For the criteria B, the species can be assessed as endangered under criterion B1 plus B2 AB3. This criteria is based on EOO less than 5,000 kilometers square, AOO less than 500 kilometers square, number of location is less than 5, and also continuing decline in area, extent, and quality of habitats. For the criteria C, the species is also reassessed as endangered under criterion C2A1, which is based on the number of mature individual less than 2,500 individual, continuing decline in area, extent, and quality of habitats, and also total number of mature individual less than in each subpopulation is less than 250 individuals. For the criteria D, the species can be assessed as VUD1 as the number of mature individual is estimated to be less than 1,000. And for the criteria E, the last criteria, the quantitative criteria, the species can be assessed as data deficient as we have no detailed information for the species. For the conclusion, the species, as it has to be assessed with the highest category, the species can be assessed as endangered based on B1 plus B2 AB3 criteria and also C2A1 criteria. So for the conclusion, a total of 14 clans with 171 individuals was located during the survey in a study site. With additional data from other studies, the species was reassessed as endangered B1 plus B2, 3, and also C2A1. Under this category, Atelingara balikpapanensis is considered to be facing a very high risk of extinction in the wild. Therefore, immediate and comprehensive conservation action are needed in order to protect the species and its habitat. Some in situ conservation action are needed for the species, such as site management and protection, and also ex situ conservation, such as living collection at botanic garden and storing viable seeds at seed bank facilities. Thank you very much.
Thank you, Dr. Ian Robiansyah. So I think it's a very important recommendation concerning Indonesian biodiversity conservation. So I can see in the chat room there are questions for presenters, for previous presenters. So I hope Mrs. Fatima and also Mrs. Inggit Astuti can open their chat room so then they can proceed with discussions in the chat room. Right now, we will proceed to the next presenter. So Dr. Ian presented biodiversity in Kalimantan, and now we also still stay in the island in Kalimantan. We will welcome Mr. Izhar Khairullah, and he will present his presentation via video about characteristics of local rice varieties of tidal swampland in South Kalimantan. So the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. I would like to present our paper entitled Characteristic of Local Rice Varieties of Tidal Swampland in South Kalimantan. I am Izhar Khairullah with Muhammad Saleh and Mawardi from Indonesian Swampland Agricultural Research Institute, Banjarbaru. Outline of our paper: Introduction, Local Varieties of Tidal Swamp Rice in South Kalimantan, local rice varieties, characteristic of tidal swampland, and conclusion. Introduction. Local varieties still dominate paddy field in tidal swampland in South Kalimantan. Definition: local varieties, long duration, low yield potential, less fertilizer and pesticide, adaptive to stress environment. Farmer have long planted various local rice varieties in tidal swampland of South Kalimantan. The varieties of Siam, Bayar, Pandak, and Lemo are well-known local varieties. This paper is a review of studies related to local varieties rice of tidal swampland in South Kalimantan. And the aim of the paper is to describe characteristic of local varieties of tidal swampland in South Kalimantan. Local varieties of tidal swampland in South Kalimantan. Paddy fields that had been opened were originally called Bayar field because the rice varieties planted were the Bayar variety. Segregation of Bayar variety soon up name of new local varieties such as white Bayar, yellow Bayar, and melintang Bayar. The naming was related to the color of light yellow or straw yellow. The description of Bayar variety, long duration, 10 months, nursery, 4 months, photoperiod sensitive variety, sensitive to water stress, easy to stress and fall, not drought resistant, plant head tall, grain yield was 2.5 to 3 ton per hectare. The segregation of local Siam variety such as Siam halus, Siam menengah, Siam Ghana, and the segregation of Siam unus variety to Siam unus halus, Siam unus kuning, and Siam unus putih. The name refers to shape and grain color. The characteristic of local rice varieties of tidal swampland, agronomic trait and morphological characters such as lip angle, plug lip length, lip width, calm angle, tiller number, ligule shape, grain, panicle, and brown rice. Some agronomic traits of local rice varieties in tidal swampland of South Kalimantan, such as Siam unus, Pandak, Bayar Palas, Lemo Kuatif, and Lakatan Gadur, with agronomic traits, tiller number, duration, lip width, calm diameter, grain length, and lodging. The characteristic of grain. Fa and Zn content of local varieties were Siam Pandak, so iron content highest, 83% ppm FA, but for ZN content, the highest shown by Siam Pananga with 110 ppm ZN. Some local varieties resistant to disease and insect of several local rice varieties, such as brown plant hopper biotype 1, resistant blast lab rice 2, and moderately resistant to brown spot. The characters of tolerant to iron toxicity. We were testing in acid sulfate soil with FA content was 156 ppm FA in soluble soil. FA concentration in groundwater at first higher, then decrease with time. The FA toxicity symptom indicate that there were considerable variation between seedling old. Seedling of 1 week old showed more resistant than seedling of 2 week and 3 week old. Siam perak and siam randah putih show the tolerant FA toxicity for 1, 2, and 3 week seedling old. Conclusion: agronomic traits of local varieties were observed. The learning ability, panicle exertion, lodging, grain weight, grain per panicle, and grain year per hill. Morphological characters such as leaf plate, ligule shape, panicle length, grain length, militarized performance. Iron and zinc content of brown rice. Siam Pandak variety contain 83 ppm at A, while Siam Pananga contain 108 ppm ZN, resistant to disease and insect of local rice varieties such as brown plant hopper biotype 1, leaf blast rust 2, and brown spot. Tolerant to iron toxicity such as shown by Siam perak and Siam randah putih. Of the conclusion, some of local varieties could be used for the breeding program for improving new high-yielding varieties of rice. Thank you for your attention.
Thank you very much, Mr. Izhar. Before we continue to the next presenter, I have an announcement. So there will be best presenters in this Isibio, and it will be announced in day 3. So stay tuned in Isibio and please follow it daily until day 3, until 20th of November. So right now, we will proceed to the next presenter. We will hear again about a research conducted by Dr. Yuni Setio Rahayu and team. So previously we hear about research in Mount Gede Pangrango, and right now it's in the neighboring national park. Yeah, she will present via video also about the composition rate of some dominant tree species in low montane forests, Halimun National Park, West Java, Indonesia. The time is yours.
Thank you, Moderator. Ladies and gentlemen, good afternoon. It is a good opportunity for me to deliver a research presentation with the title Decomposition Rate of some Dominant Tree Species in Low Mountain Forest of Halimun National Park, West Java, Indonesia. My name is Yunis Rahayu from Research Center for Biology. Ladies and gentlemen, it is well known that the nutrient cycle is important cycle in the forest ecosystem, and the return of nutrient in the forest ecosystem depends on the regular transfer of nutrient between components, and one of the main links of this transfer is litter flow. Nutrients stocked in the forest biomass return to the forest soil through the litter flow. And litter flow is the principal source of organic matter and starting point in a pathway of the transferring organic matter and chemical elements from the vegetation into the soil surface. And the nutrient mineralized during the decomposition process of decomposed litter varied among tree species. Those nutrients are important for the production of biomass and the plant growth. Nutrient release from the decomposed litter, important source for primary production, particularly in the tropical forest, which is well known to have poor soil nutrient, since the nutrients release vary with the forest type, species, stand attributes, and the variation of environmental condition. Studies on the release of nutrient by decomposition have provided diverse results even for litter of the same species. And the objective of the study are to examine decomposition rate of leaf litter and to investigate the role of some dominant species contribution to nitrogen and carbon input in the forest. Our study site was in Gunung Halimun National Park, West Java. First plot located in the ridge north of Gunung Kendeng near Cikaniki Research Station, named with plot 2. The dominant species in this plot is Altinga excelsa, and subdominant species is Simawalichii. In the second plot was located in Wates area, plot 3, near the border of Bogor and Sukabumi district, about 2 kilometers from plot 2. And the dominant species is Castanopsis acuminatissima, and Simawalichii as the subdominant species. Litter bug experiments were used to estimate the decomposition rate of, in the forest floor. And the estimation of the dominant species decomposition rate was conducted by monitoring the weight loss of litter from the litter bugs. And the litter bug techniques is the most standardized method for studying early stage of litter decay. Litter fall from the litter trap was separated into 5 dominant species and the other as mixed litter. Leaf litter were then air-dried and placed into litter bag made of nylon with a mass size of 2 millimeters. Each bag contained 10 grams of litter. And before setting of litter bag, the litter on the ground surface were gently removed to create the direct contact to the bugs and soil surface. And each location for litter trap were collected for each species at every sampling time, with the total litter bag were 200 bags. This is the formula to estimate the decomposition rate. Now we are going to the result and discussion of the decomposition rate of some dominant species. From this set shows the decomposition rate of the dominant species in Gunung Halimun National Park higher in the plots 2 compared to the plots 3. It is predicted that the stable soil temperature in the plots 2 maintaining high level of microbial activity in the soil and leads to the high litter decomposition rate. And the higher weight losses of leaf litter in the plot 2 are possible consequence of moisture condition. If we look at the lignin and nitrogen's content of litter of some dominant species, it is well known that litter quality is one of components that can control litter decay rate. And lignin, nitrogen, carbon, and phosphorus have been known as indicator of litter quality due to their influence on litter decaying rate. And the combination of high nitrogen and lignin content of Castanopsis javanica and Quercus lineata show its high decomposition rate compared to Althenia excelsa and Simawalichii. In this figure indicate with the red circle with the square and triangle signs. Althenia excelsa with the highest lignin content was decomposed slower than other dominant species in both study sites. In this figure, indicate with the green square. And the combination of low lignin content and high nitrogen content of Quercus lineata gave results of high decomposition rate. Those rate almost similar for Castanopsis javanica. This could be explained that the litter quality is one component that can control litter decay rate, lignin, nitrogen, carbon, and phosphorus have been known as indicator litter quality due to their influence on litter decay. Now we are going to the discussion of the nitrogens, carbon, and CN ratio of leaf litter during the decomposition process. The nitrogens and carbon content pattern was almost similar for 5 dominant species in these 2 locations. The nitrogen and carbon content increased from October to December, and then decrease slowly or relatively remain constant until the end of the study period. This indicated by the red arrow in the left and the middle part of this slide. And if we look at the CN ratio pattern, in both plot decrease faster during September until November, and then slowly decrease until the end of the study period. Decrease pattern of CN ratio was almost the same for all the dominant species, and the CN ratio of litter from dominant species reflected by decomposition rate of those 2 species, which they were being the species with the highest and lowest decomposition rate. Now I will explain about the combination of biomass production from the litter fall and its nutrient return by comparing based on several literature. In this table, we try to compare some study of biomass and litter production. This research shows that litter biomass on the forest floor of Gunung Gede Gunung Halimun National Park was lower than other forests. High amount of annual litter fall in Gunung Halimun National Park resulted in a lower accumulation of litter on the forest floor due to the high rate of decomposition and leaf turnover rate. This suggested that nutrient cycle is faster in the Gunung Halimun National Park than that of in the heath and peatswamp forests. While we compare the annual litter fall rate was higher in Halimun National Park than in the mixed diptero-carp forest, resulting in less leaf litter on the forest floor, but similar turnover rate. Furthermore, rate of nutrient cycles in the study's forest was higher than in the mixed diptero-carp tropical forests in Borneo. It can be concluded that the nutrient cycles in Gunung Halimun National Park rapidly than other ecosystem types in Indonesia. In conclusion, based on the study, it is concluded that the rate of litter decomposition of Quercus lineata was higher than other species due to the high nutrient content, while the lowest decomposition rate was documented for Altingia excelsa, and the carbon and nitrogen release pattern was almost similar for the 5 dominant species in the 2 locations. The nitrogen release of 3 dominant species was occurred at the first until the 3rd month of incubation time and gradually released until the end of the study period. And based on the rate of decomposition, it is predicted that high litter decomposition rate of dominant species contribute to the rapid release of nutrient in the forest output and good location. Thank you very much.
Thank you very much, Dr. Juni. So we will have 2 more presenters, and as I mentioned to you before, there will be a best presenter chosen from this session, and it will be announced in day 3. Okay, for the next presenter will be Mr. Muhammad Yunus, and he will be presenting mapping of quantitative trait loci for yield component traits from an Indonesian rice local variety, untuk Rajab. So the floor is yours.
Assalamualaikum warahmatullahi wabarakatuh. First of all, we would like to thank the committee for giving us this opportunity to present the results of our research. And the title of our presentation is Mapping of Quantitative Trait Loci for Year Component Traits from an Indonesian Local Rice Variety, untuk Rajab. And the member of the team are myself, Muhammad Yunus, Adani Satyawan, Ahmad Adang, and Ahmad Warsun from BB Biogen. As we already know that rice is a staple food in Indonesia, and demand for rice increase every year because of the population growth, and developing rice varieties for high yield is an option that can be used to address the situation, and genetic variability is an essential for developing new rice varieties. And Indonesia has rich rice gene platform comprising local varieties and wild species. And local varieties has high genetic variability that can be used to identify genes related to your component traits. And untuk Rajab is one local rice variety originally from Banyuwangi. This variety has resistance to brown plant hopper, and we noticed that this variety has high percentage of MTC that can be used to identify QTI related to yield component trait. That's why using this variety, the objective of our study was to identify quantitative trait loci related to yield component traits, such as panicle number per plant, total spikelet per panicle, field spikelet per panicle, empty spikelet per panicle, and ratio of field spikelet to total spikelet number per panicle. In this study, we use 114 individual F2 plants generated from a cross between TN1 as the female parents and untuk Rajab as the male parents. And each individual F2 plants was grown in a pot in the greenhouse. So this is the picture of F2 plants with the both parents. And to obtain the phenotype data for each year component traits, we evaluate panicle number per plant by counting all panicle per individual plant, and for total spikelet, field spikelet, and empty spikelet per panicle as average of counting the spikelet type in the 5 first emerging panicle per plant, and for the ratio of a field spikelet to total spikelet number per panicle by dividing of field spikelet number by the total spikelet number per panicle. And for genotype data, we isolate the DNA from leaf of each individual F2 plants using Delaportas methods, and positive analysis was conducted at diri service laboratories. And for genetics map construction and identification of QTL, we use those phenotype data and genotype data. And for genetic map construction and identification of QTL, we use inclusive composite interval mapping method in QTL IC mapping software, and use LOD threshold 2.5 for QTL identification. And we obtain results such as phenotype data for empty speculate for panicle was skewed and need to transform into logarithmic form, and also for ratio of field speculate to total speculate for panicle was transformed into accent form. And for genetic linkage map, we use 885 polymorphic markers for genetic linkage map. It was about 73 markers in average per chromosome, with the total length of the genome map was about 4,000 centimorgan, with 4.6 centimorgan distance in average between two markers. And this is the map of the location of QTL in chromosome. And we found that no QTL was identified for panicle number per plant. This is the more detailed information about the location of each location of each QTL for your component traits, and such as the location of the chromosome and the flanking marker and the phenotypic variance effect value. And for the total spikelette number, we found two QTL in chromosome 6 and 9, and for field spikelette number, we found 3 QTL in chromosome 4, 6, and 12, and for ratio of field spikelette number to total spikelette number, we found 2 QTL in chromosome 3 and chromosome 12, and for empty seed, we found only 1 QTL in chromosome 11. And we also try to compare the result of this study to previous study, and also the information from QTARO gene browser. And we found for spikelette number per panicle in previous study, QTL detected in chromosome 4, 5, 6, and 12 by several researchers. And QTARO gave information that in chromosome QTL in chromosome 6 and also in chromosome 9 related to spikelette number per panicle. And for field spikelette number, previous study found QTLs in chromosome 1, 2, 3, 4, 6, 7, 8, 11, and 12. And then QTARO also give information QTL in chromosome 4 and 12. And for ratio of field spikelette number to total spikelette number per panicle, QTARO give information QTL in chromosome 3 and 12 as well. And the last for empty spikelette, the previous study identified QTL in chromosome 2, 3, 11, and 12, and QTARO give information QTL in chromosome 11 related to floral organ number. However, further studies are still needed to confirm the utility of identified QTL with high phenotypic variance effect values, and to demonstrate that the new candidates QTL would be beneficial for improving rice varieties. And then we conclude that no QTL was detected for the number of panicles, and 2 QTLs were identified for the number of spikelette per panicle in chromosome 6 and chromosome 9. 3 QTLs were identified for the number of field spikelette per panicle in chromosome 4, 6, and 12. 2 QTL were identified for the ratio of calculate to total spec number for panicle in chromosome 3 and 11 and 12, sorry. And one QTL was identified for the number of empty speculative panicle in chromosome 11. I think that's all for my presentation, and thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mr. Muhammad Yunus. So I can see there are many question and answer going on in chat room. So we will have our final presenter in this session. So if you still have question for the previous presenters, you can write the question in the chat room. And please, for the previous presenters, you can open your chat room in Zoom and answer directly there for the question directed to you. Now, for the final presenter in this afternoon session, let's welcome Mr. Kurniawan. He will present via video about bioresources and diversity of snakehead, Canastriata, a proposed model for optimal and sustainable utilization of freshwater fish. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh, everyone. I'm Kurniawan from Research Institute for Freshwater Aquaculture and Fisheries Extension. On behalf of Professor Rudi Cristiano and Iriana, I would like to share our research project. It's about the bioresources and diversity of snakehead, Canastriata, a proposed model for optimal and sustainable utilization of freshwater fish. So snakehead is an essential and valuable species in fishery sector, usually used for food consumption, ornamental fish, and pharmaceutical material, especially for albumin resource. So snakehead production from Indian fisheries still dominate the production. It's around 92.8%, and the remaining from aquaculture. So if the situation occur, the wild bioresources of the snakehead will be threatened due to overexploitation. On the other hand, snakehead aquaculture usually still use commercial feed as the main input of the production, but it can release the organic waste, uneaten feed, and nutrient to the aquatic ecosystem. So optimal and sustainable utilization of these excellent bioresources is very essential to improve snakehead production. So the aim of the research is to discuss snakehead bioresource and diversity, existing production, and strategies for optimal and sustainable utilization of the snakehead. So the method used for this research, we try to investigate the snakehead breeding technology in our farm and the hatchery, and we have a global production workplace for fish statistics, and we combine with national fisheries statistic database, continue to conducting critical review in identification model of fisheries management. We decided that the application of bioresource flow model for integrated agriculture and aquaculture system to improve sustainable utilization of bioresources is very important. Diversity and potential bioresources of snakehead, from the identification of the species inhabit the freshwater ecosystem in Indonesia. The snakehead have 10 species, but from the 10 species, there are only 2 species usually used for fish consumption and albumin resources. The remaining of the snakehead usually known as the ornamental fish. So we still have many resources for development of the snakehead, for albumin resource, or for the fish consumption. So the trend of snakehead production in the world, from inland fisheries and aquaculture, we have two graphs here, and two graphs, the production from inland fisheries still dominate the production of the snakehead. Similarly, in Indonesia, snakehead from aquaculture still less than around 10%. The remaining is come from inland fisheries. The production in regional area, Kalimantan and Sumatra, is the largest producer of the snakehead, followed by Java and Sulawesi. And the aquaculture system used to cultivate this fish usually use ponds around 80%, followed by paddy field 13%, and the cages around 7%. We have challenges in snakehead production, both in aquaculture and inland fisheries. In aquaculture, we have three challenges in spawning, larva rearing, and juvenile rearing. These activities still depend on the seasonal condition. For example, snakeheads only spawn in rainy season. So for the production, usually we use hormonal treatment to maturate the gonads. In inland fisheries, water temperature is very essential for snakehead breeding. So it will depend on the production will depend on the breeding naturally in wild area. So we use bio-research flow model for optimal and sustainable use of the snakehead. Bio-research flow model present interaction of the essential component and the price through the integrated system considering biological, economic, and environmental aspects. For example, we have 4 components. The main component is related to the other one. So, for example, for the aquaculture development, the first step, we need to domesticate the wild fish in the captive area. So as the domestication succeeds, we can continue to hatchery production for produce the seed that can use for growth in aquaculture ponds, or we can restock in the natural freshwater area. And the seed can also be used for ornamental fish. And for aquaculture production, we need around 6 to 1 month to get the commercial size of the snakehead, and we can continue to produce the fish based on biotechnological processing, such as we can create albumin and creatine from the snakehead, and we can use conventional processing to produce dishes or fillet, like fresh, frozen, smoked, and salted fish. The other one, we can continue to alternative aquaculture production system for improvement of snakehead production. We have technology in the growth of the fish. such as biofloc technology that can be used for increasing production of the snakehead, and then the use of probiotic for improvement the production, as well as like recirculating aquaculture system for development of the snakehead production. After they succeed in the aquaculture, usually they produce the residual like ammonia or uneaten feed or the waste that can be treated for fertilizer using wastewater treatment management. So this process will produce the zero waste, and it will be sustainable use for aquaculture development and also conservation of the natural water area. So the conclusion and recommendation: first, China Sea Data is a very productive aquaculture commodity for fish food, ornamental fish, and other marine resources, but it needs an excellent strategy to take over the supply of the species from the captive fisheries to aquaculture. Improvement breeding technology is also essential for increasing seed production and maintaining sustainable production flow through integration of the sector to other sector is very essential, and the albumin must be enhanced by a new biotechnology approach and finding new prospective candidates that are more prospective in the China group. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mr. Kurniawan. So that was our final presenter for today. Thank you very much for your participation. And I would like to also announce that tomorrow we will still have many interesting oral speakers, and also on the day 3. And tomorrow we'll have 2 keynote speakers presenting. First is Professor Tomoyo Shinozaki from University of Tokyo. And then there will be Professor Muhammad Ilyas from University of Nottingham. And then on the 20th of November, which is day 3, we will have keynote speakers, Professor Peter Ng from National University of Singapore. And last but not least, Dr. Yantiati Widyastuti from Research Center for Biotechnology LIPI. And also on day 3, there will be an announcement about best presenter for the oral speaker. So stay tuned and don't forget to participate via Zoom, and also you can share this information because other participants can also join and watch your presentation from YouTube. The channel is Lembaga Ilmu Pengetahuan Indonesia. So that's all for today. But before all of us leave this room, I would like to ask all of you to turn on your video, because we'll have a photo session for this room 1. Maybe the committee can help to open the video for all the presenters and participants. Give your best smile. Okay, great.
Okay, I will take a picture 3 times because we have 3 slides. Ladies and gentlemen, please open your camera. Okay, get ready on slide 1. 3, 2, 1, smile. Okay, on the slide 2. 3, 2, 1, and smile, everyone. Last slide. Okay, 3, 2, 1, smile. Okay, thank you so much.
Okay, thank you very much. Don't leave yet, because don't leave the Isibio yet, because we will all be directed to the main room for now. So thank you very much for participating the session 1. I will say goodbye for now. See you tomorrow. Bye bye.
Ladies and gentlemen, we have followed all agenda today. Thank you very much for all speakers, moderators, distinguished guests, and participants who have attended and actively participated in the symposium. Tomorrow, we will continue our symposium. To all participants, please be aware to change your username according to the format given in the conference room. Please join on time. Thank you very much and see you in the next session. Ladies and gentlemen, we have finished for our agenda today and you are pleased to leave the meeting. See you tomorrow.